Let us go ahead and get started. So we are where we left off last time, continuing our discussion of muscle physiology, lab doing our origins, insertions, and actions, which we're a little behind on, but I'm hoping we can catch up some today. And like you, I'm sure you're very, very excited because we are off next week. So get a chance to study, get caught up, sleep, do all those fun, important things. Say hello to your family that you haven't seen in a while, and get ready for the final 2 months of this class. So again, enjoy your free time because after this weekend, your butts belong to me. Excellent. We have 3 more assignments, a PhysioX, so you're not going to be completely free during the break because, as you know, the PhysioX is not hard, but it is time consuming, and exercise 2 has 7 activities, so 7 lab reports. Luckily, you get to turn them in as PDFs online, so you don't have to kill a small tree printing them out, but make sure you do that. Thursday, the following week, your Unit 11 review and your muscle activity handout, which kind of goes along with what we've been doing in lab, is going to be due, and it will be graded for correctness. All leading up till the 13th, which is our last lab and lecture exam. And that's the last thing that I want to talk about. The other thing that's been going on over the past couple of days is I've been meeting with most of you for our midterm 5 for 5s, talking about one of the things. And one of the consistent themes that I think has come up both for, and again, for those of you who haven't, I know I've got a couple meetings tomorrow and then a couple of other open slots if anybody wants to take them, if there's anybody left who hasn't signed up. And there are still a couple of you. If you feel the need, you're welcome to do that. But one of the things that I think I've consistently been talking to people about, and one of the consistent things where people are missing out on points, is that detail. And I think that's one of the things we've really been emphasizing in the past couple of lectures. And in particular, something that we've pointed out with a lot of people is at the end of the last class, when we went through all of the steps of the relaxation of the muscle, and we put all the pieces together to see how it all works together. And I think that that was something that many people thought was useful. So I thought it might be useful for us to go ahead and do that exact same thing again, this time with the process of contracting the muscle. So if there aren't any questions before we get started, I think the best thing for us to do is actually to take the time to actually go through and talk about the process of how a muscle contracts. One of the points I want to emphasize is that there is no new information in what we're providing here. We're just consolidating all of the information that we've talked about to in the hypothetical, which totally wouldn't happen on the exam because we're going to break it up into pieces. But if on some future exam you have in some future class, someone asks you the process of how a muscle contracts, you can go through it step by step by step. So let's start easy. If we decide we want to contract the bicep brachia, how does that process begin?
Communication at the neuromuscular junction.
True, it is going to take place at the communication at the neuromuscular junction, but what is the very first step in that process? I don't want to break it up into the pieces this time. I want to go through the entire process of how it contracts. So you're right. We need to get to the neuromuscular junction and communicate there. But what has to happen first?
Make a decision.
Absolutely. Excellent. And several of you got that on there. We make the conscious decision to contract the muscle. And when we do that, we produce a neural action potential. Excellent. Let's give myself a little bit more room. I think this will all fit on the screen. We'll find out. Of course, we know that neural action potential is a big positive signal, and that big positive signal spreads down the axon. And it reaches our synaptic end bulb. Now, what is the effect of that neural action potential reaching our synaptic end bulb? What does that big positive signal do to our synaptic end bulb?
Wouldn't it open a channel?
It is going to open a channel, but why does it open the channel? What change does that neural action potential cause that leads to the opening of the channel?
Depolarization.
Excellent. It makes the synaptic embole more positive. And there you go, Ariel's got it. Exactly. The neural action potential depolarizes the synaptic embole to, or at least past, some magical point. And what was that magical point?
The threshold of -60 millivolts.
Excellent. Threshold, which is -60 millivolts. Excellent. I love that. Then, as you guys pointed out, at threshold, a channel opens. But what kind of channel opens?
The voltage-gated channel, calcium channels.
Excellent. Voltage-gated calcium channels open on the synaptic embole after we pass threshold. Again, and this is one of those things that we talk about. Just saying that a channel opens is absolutely 100% correct, right? But we want to talk about all of the immediate effects of this and all the steps and all of the details. So rather just saying that we make a decision to contract the muscle and channels open at the synaptic embole, we're talking about producing that neural action potential, about depolarizing the synaptic embole, reaching that critical point of threshold, and opening voltage-gated calcium channels. And of course, as soon as those voltage-gated calcium channels open, what happens?
You have an increase in calcium that triggers exocytosis.
Excellent. Calcium enters the synaptic embole, and that calcium entering the synaptic embole triggers, and I like we using that word again, but absolutely, it triggers vesicles. Vesicles containing acetylcholine to bind to the plasma membrane and release their contents via exocytosis. Excellent. Oops. And we can even be more specific than that, into the synaptic cleft. Excellent. What happens once that acetylcholine enters the synaptic cleft?
It diffuses across the synaptic cleft into the motor end plate.
Excellent. So it diffuses across the synaptic cleft to the motor end plate of our skeletal muscle cell. And what happens when it gets there?
It opens channels.
Excellent. And again, good, correct information, but can we be more specific?
The acetylcholine binds to chemically gated cation channels and opens them.
Absolutely. Binds chemically gated cation channels and opens them. Excellent. When that acetylcholine binds to chemically gated cation channels, some cations are going to move. What cations?
Sodium and potassium.
Excellent. What does sodium do?
It
There we go.
Depolarizes.
Ariel's got it. Enters the cell, and potassium leaves the cell. Excellent. So those are the two things that are going to occur. However, do they occur equally?
No, there's more sodium.
Excellent. So lots of sodium enters into the cell, and only a little bit of potassium leaves the cell. And what is the net effect of this unequal movement of cations?
The motor end plate gets more positive.
Yeah, so the net effect is we get a depolarization, or you could just say it gets more positive, absolutely, depolarization of the motor end plate. Now, if that depolarization is big enough, and again, that's a key part of it, this isn't an always happening type of event, but if the depolarization is large enough, and how do we know if it's large enough?
Because it reaches its threshold.
There you go. If it's large enough to reach threshold, then something magic happens. What's that something magic that's going to happen?
We
There's some
generate
action potential.
a muscle action potential.
Exactly. We're going to reproduce a muscle. Oops. Action potential at the motor end plate. And then what happens?
The muscle action potential spreads down the sarcolemma.
Excellent. Then what happens?
The calcium channels open.
That is going to happen, but something's got to have something. Where are those where are those calcium channels located?
On the terminal cisterna.
Of the sarcoplasmic reticulum, and those aren't on the surface. So you guys have the right idea. We need to open those calcium channels, but we got to get that signal from the sarcolemma on the outside down into the cell where those terminal cisternae are located. And there you go. Ariel's got it. Our muscle action potential spreads down the sarcolemma, and our muscle action potential spreads down the transverse tubule. Of course, when it spreads down the transverse tubule, as you guys pointed out, it is going to depolarize the T-tubule, and the two terminal cisternae next to it that together form what structure?
The triad.
Triad. Excellent. And then you are correct. When those, I think I got room for one more spot here. When those depolarize, and again, to threshold, what happens?
Then the voltage gated channels open.
Excellent. Voltage gated, what kind of channels?
Calcium channels.
Calcium channels open on the sarcoplasmic reticulum. Oops. Excellent. All righty. What happens next?
This causes calcium to go out into the cytosol or the sarcosol.
Yeah, no, cytosol is fine. Absolutely perfect. Into the sarcoplasm or the cytosol, absolutely. So calcium exits the sarcoplasmic reticulum, which I just finished spelling out. So again, I can abbreviate it, into the cytosol of the cell. Excellent. And what happens to that calcium?
The increase of calcium causes the calcium to bind to troponin.
And what happens when calcium binds to troponin?
It undergoes a conformational change and turns and rotates the tropomyosin.
I just love saying undergoes a conformational change. It sounds so grown up, but you guys are absolutely right. It just means it changes its shape. And when it changes its shape, the troponin rotates the tropomyosin. Protein. And when that rotates,
the now active sites on actin are available, and the myosin heads can grab the actin site to start the contractile cycle.
excellent. So rotation exposes the active site on actin, and now primed myosin heads, right? It has to, we know it has to be a primed myosin head, and bind to the exposed active site.
What does primed mean?
You tell me, what does it mean for the myosin head to be primed?
I don't remember.
So somebody else, help her out.
It's faced away from the M line.
Excellent. So and again, one of the things to remember when we talk about a cycle, it is a continuous process. So we could actually put that in here. So let's go ahead and do that. If it has to be a prime myosin head, we need to prime the myosin head first. How do we prime? How do we prime a myosin head?
By splitting ATP into ADP and phosphate.
And energy. And remember, it stores all three things. And in that process, it turns the head. Excellent, right? So there we go. This part right here is a perfect thing to put in this spot right here. We can do that. Okay, let's go ahead and do this just to, perfect, because we'll see where else we could have put that as well. Putting it here is perfectly acceptable. So we prime our myosin head. Myosin head splits the ATP and ADP phosphate and energy, stores all three of those things so that it is pointed away from the M line now. And now, as you guys said, that prime myosin head can bind to the exposed active site on the actin. And what do we call that process?
The cross bridge.
There you go. Forming a cross bridge. What happens once that cross bridge is formed?
The myosin head can release its energy.
Uses the energy to do what?
To perform a power stroke towards the M line.
Why is that pivot towards the M line so important?
It helps contract the muscle.
Yeah, how? When it pivots towards the M line, what does it actually do?
Does it shorten the microfibril?
Yeah, myofibril, absolutely. Right, but let's walk through it. When it actually pulls, what it actually does is it pulls the actin towards the M line, which of course pulls the Z disc towards the M line, which makes the sarcomere shorter. All right. Because that sarcomere is connected to the rest of the muscle cell, it makes the whole myofibril shorter and makes the muscle shorter. I'm not going to be able to fit everything in if I leave these all on separate lines. So let's cheat a little bit. Pulls actin towards the M line, which pulls the Z disc towards the M line, makes the sarcomere shorter. Our myofibril gets shorter. Our muscle gets shorter. And this is where we generate our force, produce our tension. Excellent. Now, the myosin head does one other thing while it's performing that power stroke.
Is it releasing the ADP and phosphate?
Yep. Exactly. While performing that power stroke, it releases the ADP and phosphate. All right. That's good because once it's done, it now has an open binding site. So after the power stroke, keep this going, what has to happen?
A new ATP binds to the myosin head, causing it to let go.
Absolutely. After the power stroke, a new ATP binds to the myosin head. That causes it to let go. Just say it that way. Binding of the ATP breaks the cross bridge. And the myosin releases the actin. Then what happens? Is the myosin head ready to do work?
Not yet.
No. Myosin head must split the ATP to become primed again, or revitalized, or cocked, or whatever term you want to use. And the process continues. Notice what that means is this part right here about how you prime a myosin head, we could have just as easily put right there. Because it's a cycle process, it could either be the very first step or the very last step, and both would be a fine way to talk about it, because either way, we're priming that myosin head and we're ready to continue and we're ready for it to do work again. Okay, so does that make sense? So if you wanted to prime it first, that's great. If you wanted to start with the primed head and then reprime it by explaining that process at the end, either way, that would be fine. And so just that simply, we have gone all the way to the point where we made the decision to the point where the muscle contracts. And I know it is impressive to look at on the board, but as we've talked about, there is absolutely no new information on the board here. This is just all the information that we've talked about. But as you can see, it's not a hard process, it's a long process. So to make this long process more digestible, how many pieces did we break it up into?
I thought 4.
Well, but what we're seeing on the board, how many processes is this here?
3.
A lot.
3. It's just 3 processes, absolutely. What was the first process?
Communication, the neuromuscular junction.
Communication at the neuromuscular junction. Excellent. And if instead of asking you exactly how a muscle contracts, I asked you to describe initiation of communication at the neuromuscular junction, where would that have started?
Making a decision.
Excellent. And where would it have ended?
The end bulb.
Where
A little
the
further than the end bulb. Remember, what's the goal of communication at the neuromuscular junction?
to create the muscle action potential.
So where does it end?
Down where the, right there.
There you go. Where we produce a muscle action potential at the motor end plate. Or yeah, you could even say where it spreads. I'd be fine with that. Either of those are acceptable. What was step 2?
Excitation, contraction coupling,
Where does that start?
where it spreads down the sarcolemma.
Yeah,
Or-
again, producing the action potential, it's spreading down the sarcolemma. Where did that end?
When the myosin heads can grab the actin.
Excellent. So how about when troponin rotates the tropomyosin protein, exposing the active sites of bind so that the myosin can grab onto the actin. Excellent. And what was step 3 in our process?
The myosin heads binding to the active sites.
Wait, what? So yeah, but what's that third step called? What's the third step in our contraction process called?
The contractile cycle.
Contractile cycle. Excellent. Where does it begin?
Binding to the actin.
Well, really, we have to expose the binding site first, right? Remember, the end of one step is the beginning of the third. And technically, does this process end?
No.
No, technically it continues, but at least our description of it basically stops there. So there you go. What we have here are the three processes that we've already learned. And now we see that it really is one continuous process from the decision to the point that a muscle contracts. Those are all the pieces we have to put together to make sense of this. All right. All the steps, all the details, all the material. This is the kind of thing that I'm looking for on the exam. Excitation, contraction, coupling.
So in that second, in the second step, do we need to talk about calsequestrin at all or not particularly?
No, when the same way, notice we didn't talk about any of the calcium pumps for this one as well, because in the initiation, they don't play a role.
Okay.
When we're terminating it, that's where the pumps make a difference. That's where the calsequestrin makes a difference, because it's one of the things that lets us store all that calcium in the sarcoplasmic reticulum. So yeah, those are things that we talked about that they were there, but they're not playing. Notice we didn't talk about acetylcholinesterase right here either, because they're not playing a role in this process.
Okay, I had it in my notes, but I didn't have any details about it, so I just wanted.
Right. Well, because when we talked about the anatomy, we know those things were there, but you know, because they're going to play a role in the termination of it, not in the initiation of it, which is what we have here.
Got it.
Okay. All right. I think that hopefully that is useful. That was useful when we did it. I think a lot of people appreciated when we did that for the muscle relaxation. So I thought it was worthwhile to do it for the muscle contraction. And again, your book's got all the pretty pictures and stuff like that. Yeah, I think I said that, right? Excitation, contraction, coupling. Some pretty pictures showing this as well. Again, from the excitation to the muscle action potential, to the moving of the regulatory proteins, to the contractile cycle. Excellent. And even though we talked about it in the last class, let's emphasize it again. What determines how long a muscle is going to contract for? There's really technically three things. So what are the three things that you think are going to play a role in determining how long a muscle contracts for? What are the things that are needed?
Resources available.
ATP.
Okay, and so one of those resources is ATP. Excellent. What's a second important resource that must be present?
Would it make sense how long it keeps continuing to get the message that it should contract from our brains?
Absolutely. So notice another big factor is going to be, right, how many or how long we produce neural action potentials. And notice how many neural action potentials we produce lead to the release of acetylcholine. So how much acetylcholine isn't present because of either how many action potentials neural action potentials we're producing, or right, if we have more enzymes in the synaptic end bulb, it's going to get broken down. If the cells are further apart, they're going to diffuse out more rapidly. So yeah, so how much acetylcholine is there? The amount of acetylcholine. So let's say it that way. The amount of acetylcholine, the amount of ATP, and what's the other thing?
Calcium.
Yeah, there you go. Right. Those are the three main factors that are going to determine how long we're able to contract a muscle for. Yes, Yulia.
So acetylcholine, do we have only a limited amount of it? Or how does it get produced?
It is a protein that is going to be constantly made by the neuron. So it is going to be made in the cell body of the neuron and shipped down to the synaptic end bulb. And remember, some of it will be endocytosed so that we can recycle it and use it again. But yes, how much acetylcholine you have to release is something that is going to be modifiable in a couple of ways. How many vesicles bind and release? Right, the more vesicles that bind and release, the stronger the contraction, the longer the contraction you're going to get. How much acetylcholine you put into a vesicle, right? The more you use that connection, the more acetylcholine will make, and so the more will bundle up. So when one vesicle releases, instead of releasing 10 acetylcholine, it can release 20, and you can get a much bigger effect. And like we said, we can affect how close they are together, how much invaginated the motor end plate is, how much enzymes we have in there. All of those are things we can use to determine how long that acetylcholine is. So really, when we talk about that stimulation at the neuromuscular junction, like you said, we're really talking about the amount or how long that acetylcholine is going to be in the synaptic cleft. But we also have to have calcium to move those regulatory proteins. And we need to have ATP so that the myosin heads can do their work. Absolutely. So all of those are factors that are going to influence it. All right. Now, as we mentioned, these types of contractions cause the sarcomere to shorten. And we can actually see that when we look at it here in an illustration and see it actually under the light of the electron microscope as well. Notice, oh, hold on, let's go back. There we go, the picture you want. If I remember correctly, when we talked about the regions of the organelle, that is the myofibril, can someone identify me the regions for it again? What were some of the regions we identified? If you're not sure, you can cheat and look at the picture right next to me.
The Z line, A band, I band.
Yeah, exactly. One is the A band. Excellent. One is the I band. Excellent. What else is there?
H
M line. What else?
H
E
band.
line. Excellent. And the H band. Now, as we look at the relaxed sarcomere versus the contracted sarcomere, do any or all of these change, or do just some of these change?
I think they all change.
Well, let's figure it out. Does the A band change?
Yeah.
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 Or
How?
no.
Does it get
No, it
longer?
doesn't.
Does it get shorter? What happens to the A band?
It gets shorter.
How do we define the A band again?
The
It's
darker region that carries the thick filaments?
Yeah, it's the region that is defined by the myosin. Is our myosin changing length? Is it changing shape?
No.
no.
No. The myosin heads are grabbing and pulling, but the actual length of the myosin fiber doesn't change. So guess what? Our A band is not changed by the contraction. But what about the I band? Does it change?
Yes.
Yes,
Because
what-
toxins on each side are.
Yeah, exactly. It gets smaller. And the reason it gets smaller, yep. Well, the M line is the center part for, oh, the M band, I see what you're saying, yep. It does get smaller because notice there is more overlap between the myosin and the actin. Remember, the I band is the region that doesn't contain myosin. So notice up here in the relaxed one, it's from here all the way over to the next myosin over here. Whereas notice here, those two myosins are much, much closer to each other. There's much more overlap with the actin and the myosin. So as a result, our I band gets smaller. All right. What about the M line? Does the M line change?
No.
No.
It is, or let's be specific, is not changed. It stays smack dab in the center. But what about the Z lines? Do the Z lines change?
Yes.
Yeah, they get closer to each other. And what about the H band? Remember, the H band is the region in the middle that just contains myosin. Does our H band change?
Yes.
No.
Yeah, it gets smaller.
Yeah,
Smaller.
it gets smaller. And in fact, it is actually possible for the sarcomere to contract enough where the actin from one side would overlap with the actin of the other side. And if that occurred, what would happen to the H band?
Would
Disappear.
Disappear. Exactly. If it from both sides came together, it would go away entirely. So it absolutely can change. Now, it's easy to see this and think about this on a simple illustration like this. But remember, as we've seen, we can actually see the organization of these proteins with an electron microscope. So again, notice in our electron microscope here, this great view, we can clearly see where the Z discs are. We can clearly see where the myosin is for the M line. We can see the portion where the myosin and the actin are overlapping. And we can see the part that just contains myosin, the H band. And then we can see the part that doesn't contain any myosin, the I band. We can see that organization really, really nicely in a fully relaxed sarcomere. But the other thing we can do is before we freeze the muscle and cut the muscle and put it on the slide and bombard it with electrons, we can actually contract that muscle first. And let's actually do this. Here is the Z line. Here is the M line. Here is the H band. Here is the A band. Because what we can do is actually look at it in a muscle fiber that is contracted. Notice, as we expected, the Z discs move closer to each other as the sarcomere gets shorter. Our M line itself is unchanged. Our A band itself is unchanged. But notice that our I band has gotten significantly smaller. So these things that we speculated at don't know what that is, on the illustration can actually be seen using an electron microscope. We can actually see these changes between our sarcomere relaxed and contracted, which is, again, where we get the idea of the sliding filament theory. We can see that the myosin and actin are overlapping much, much more. Look at the difference in size of the H zone. It's basically tiny. It's almost gone away entirely because these two are sliding against each other. Yeah, you could argue that there's no, I mean, I can see a teeny bit to the side. If you notice, if I clear my drawings, you can see there's a little bit of lightness right here to the side, right here to the side in between those. So it's not quite the exact same size as the M line, but it's pretty darn close. So I wouldn't say it's gone away entirely. I would say it's practically gone away, if not gone away entirely. And it definitely can, like the illustration shows. Notice in the illustration, with the overlap of the actin and the myosin, the H band does go away entirely, and that is absolutely positively possible to happen. All right. Questions on that? All right. Now, we've done all this work to understand not just the anatomy, we've broken it down to the anatomy of the basic proteins, but now we've actually seen how the proteins work together to change the shape of a single sarcomere. And now finally, we can start building back up. And that's our goal from today. Is today we are going to start building back up. Because while we've seen everything that goes on in a single sarcomere, we know that all the sarcomeres in a myofibril are all going to do this at the same time. So notice when all the sarcomeres in a myofibril all contract at the same time, our entire Myofibril gets shorter. And as we talked about, because of myomesin, because of other structural proteins that attach the myofibril to the plasma membrane of the cell, when the myofibril gets shorter, the muscle cell gets shorter as well. And we can go even further than that, because we know that one muscle cell is bundled together with a bunch of his buddies by connective tissue into a fascicle. So when a single muscle cell contracts, the fascicle contracts. We know that fascicle is bundled together with other fascicles by the epimysium forming the muscle, so that when the one sarcomere contracts, right, the one muscle cell contracts, the one fascicle contracts, and the whole muscle shortens and changes shape, producing that tension, moving our body through space. And so those tiny little proteins interacting inside of every single muscle cell is what ultimately leads to us moving our body through space. All right? Notice whether it is structural proteins connected to the plasma membrane of the muscle cell, or then that muscle cell and its connective tissues that are connecting it to its neighbors, connecting it to the fascicles, connecting it to the epimysium. Basically what happens is the muscle cell, when it contracts, it is pulling on those connective tissues. And that is what produces our force. That is what produces what we call our tension. And it is that tension that causes the muscle movement. Right? This is physics. We're talking about a tension. We're talking about a force that is being produced. Now, when it comes to how much how much weight I can lift with my bicep brachia, right? Whether I can lift this laptop or my chair or my desk or my bed or my car, right? The total amount of tension I can be produced is going to be determined by the total number of muscle cells that are all working together. There's going to be some maximal amount of weight that from a physics standpoint, I am able to move. However, we're not quite up to the full muscle layer yet. Right now, we want to focus primarily on a single muscle cell. Right, we're no longer talking about sarcomeres. We've worked up to the level of a single muscle cell. And when we talk about a single muscle cell, there are basically two main factors that are going to determine how much tension it can produce. Because it can produce different amount of tension. If I'm going to lift this pen, do I need to use one muscle cell at its maximal tension to be able to lift this pen up?
No.
No, of course not. I can use a bunch of them at a half or a quarter of their tension and easily move this in space. So if instead I want to move our textbook, right, in that same muscle cell, I can produce a lot more tension. So how much tension is produced in a single muscle cell can be modified by two main factors. Those two main factors are actually the starting length of the sarcomere, where the sarcomere is when the contraction starts. And the second is how much input I give it from the nervous system, the frequency of the stimulation. So we are going to move up to the level of a single muscle cell now and see how that single muscle cell works, how we can change the tension in a single muscle cell. And again, one of the cool things about this is none of these are things you've probably thought of in the terms that we're going to use today. But all of these are things that I guarantee you are aware of. You've experienced it, or you've seen it, or you've taken advantage of it without fully appreciating and understanding what it was that was going on. And so these are concepts that are totally going to make sense to us as we go through them. All right. Now, it is a teeny bit early, but we have already covered a tremendous amount of information, and this is a good natural stopping point. So let's go ahead and take our first break here. We'll take our first 15 minutes break, come back at 1 o'clock, and at 1 o'clock, we will restart from here. So let me start the lecture and I will start the recording. Excellent. So let's take a quick 15-minute break, get some more caffeine. We got a busy day ahead of us, and I will see you guys at 1 o'clock. Our goal now, as I mentioned, is to start building our way back up. We've been looking at all these proteins and how they're interacting and what they're doing at a molecular level. But now we're going to start working our way back up. And like I said, our focus now is going to be on a single skeletal muscle cell. Now, as I mentioned, and actually this is probably the easiest thing to do on the whiteboard. What generates the tension of our sarcomere? My microphone's on, right? I did start talking out loud.
The myosin pulls on the actin?
Yeah, exactly. Basically, myosin grabs on the actin, it performs a power stroke, and as a result of that, that power stroke generates our tension, right? I think that's pretty simple and pretty straightforward. Let's go ahead and make a really simple myosin here. All right, this myosin has 3, 6, 9, 12 myosin heads on it, and its goal is to pull on the actin and the Z disc, right? So let's go ahead and draw one of those. There's one. There's two. And I'm going to need to cheat. There we go. Excellent. So there's a very, very basic, simple illustration of a sarcomere. Now, for this particular illustration, how many myosin heads would be able to grab and pull in this particular example?
All of them?
All of them? Well, then again, I haven't done the best job of drawing, but what if I did something like that instead? Maybe move them closer together. 8 total, absolutely, right? So notice in this particular case, and we'll go ahead and move them closer so it's more obvious. In this particular case, this sarcomere can produce 8 power strokes worth of energy, right? Well, because notice, these 4 myosin heads aren't able to grab onto the actin. So while this particular myosin filament has 12 total, right now with my sarcomere at this particular length, only 8 of my myosin heads could actually grab and pull. So I'd only be able to pull 8 out of 12 myosin heads, and so I'd pull 8 myosin heads worth of force. Now, what would happen if I stretched the myosin out? If I stretched the sarcomere out, not the myosin, the sarcomere, if I stretched the sarcomere out so the sarcomere had a starting point where it was stretched out a little bit more wider, how many myosin heads are going to be able to grab and pull now? Only 4. And notice the exact same sarcomere with the exact same anatomy, its strength has now been cut in half. From being able to perform 8 power strokes, it is only able to perform 4 power strokes. And so it is going to be much, much weaker. And theoretically, yes, Laura, go ahead. You got a question?
Yeah, I think last class you had mentioned that not every myosin head reaches at the same time, that they kind of alternately change. Is this the reason why, or is this kind of different?
Well, so you have the right idea. They are all reaching and grabbing at different times. But remember also, they're performing 5 power strokes per second. So if we're talking about a second's worth of time at the very beginning of a contraction, it's still only going to be 4 myosin heads that are going to be able to interact with the actin at this particular time. So it's, again, yes, it's going to be dynamic and it's going to change. We're talking about right at the beginning, the very beginning of this contraction, how much tension we're going to be able to produce right at the start. Yes, Yulia.
Yeah, I'm a little lost as to where we got the 5, the 8 and the 4 and the 5 per stroke.
Well, okay.
What's going on with the numbers? I'm pretty confused.
Okay, that's fine. Way back when we were talking about the contractile cycle, we talked about how a single myosin head primes, grabs, power strokes, and lets go 5 times a second. So that's where that number came from. What I'm talking about right now is the interaction between this myosin and these actins in this one sarcomere. As we were talking about, and let's write it up here so we remember, to produce tension, myosin heads must form a cross bridge with actin and perform a power stroke. All right, we comfortable with that definition? That's how we produce our tension?
Thanks.
Does that make sense? All right, I'm going to guess that means yes. So the question becomes, in my very simplistic illustration here, how many of these myosins could actually form a cross bridge, right? So notice this one can, it's right next to the actin. This one can, this one can, this one can. But notice this one's not next to the actin. These are myosin heads that are primed and ready to go, but there's no actin next to them. And if there's no actin next to them, then there's nothing for them to pull on. And if there's nothing for them to pull on, they can't do work. So that's why when we started at our original point, our normal starting point, we had it here. Notice at this point, 8. 1, 2, 3, 4, 5, 6, 7, 8. We're able to grab onto the actin, and once they grab onto the actin, they'll be able to perform a power stroke. But by stretching my sarcomere out, suddenly my muscle cell became weaker, because instead of being able to perform 8 power strokes, it's only able to produce 4. And what would happen if I stretched a muscle cell so far out that the myosin, that the myosin and the actin didn't overlap anymore? How much tension would we be able to produce now?
Almost none.
None. Exactly. And again, you may not have thought of it in these terms, but I know you're aware of this, because back in ancient times, there were these things called bars. And bars are the places you would go to on a Friday night to drown your sorrows. And some people drown their sorrows a little bit harder than other people do. And so eventually, if you hang out at bars long enough, someone's going to get unruly, somebody's going to get upset, and someone's going to get thrown out of the bar. And typically, start getting into a fight. And when that bouncer comes over to break up that fight, one of the first things they do is they grab the person's arm and they pull it behind them, right? Or if you're a police officer, for instance, trying to subdue a subject, you pull their arm behind them. Well, why do they pull their arm behind them? Because as they're stretching out the muscle, they're reducing the amount of force they can produce. And someone who's much smaller and can stabilize someone much bigger and much stronger if they stretch that arm out. By stretching that arm out, they can decrease the amount of force they're able to generate, decrease their strength, and they're able to immobilize them. So as you stretch it out, fewer myosin heads are able to grab and pull, and so strength goes down. Now, we started here with 8, but what if we shorten our sarcomere a little more? How many myosin heads can grab and pull now?
All of them?
All of them, all 12. So we're going to produce our maximal amount of strength here. Because remember, there's two things that have to happen. You have to be able to form a cross bridge, and you also have to be able to perform a power stroke. At this point, we have plenty of room to produce a power stroke. All of our myosin heads can grab and pull. So at this point, we're going to be at our maximal tension. Yes, there are a couple questions. Laura, I think you were first. Go ahead.
So correct me if I'm wrong. So when you said the sarcomere had zero tension, can you kind of relate that to someone with paralysis, or does that more of like the nervous system, or does it kind of tie hand in hand where their muscles aren't contracting?
Typically, when someone is paralyzed or something like that, it's because that, like for instance, they have a spinal cord injury. And the electrical signal cannot get from their brain to the muscle to stimulate the muscle to contract, right? So in this case, this is more about stretching that muscle out so that, you know, to stabilize the person behind you, because as you stretch the muscles out, it's weaker. And this is something you can actually experience for yourself. I'm not suggesting you wrestle your kids to the ground, but what you can do is you can have someone put their hand on your forearm, and then you try to flex your forearm. And do it in 3 states. Do it partially contracted and see how easy it is or hard it is to move their hand. Do it fully extended and see how hard it is to move their hand. And as we'll see, do it mostly contracted and see how hard it is to move their hand. The length of the muscle changes how powerful the muscle is. And that's the point we're trying to make. What can determine how much tension a muscle produces is the starting length. Because of these 2 factors, we have to be able to form cross bridges, but we also have to be able to form power strokes. So notice when we started with the sarcomere way out here, there is absolutely no overlap between the actin and the myosin. No cross bridges can be formed. All right. Now 4 cross bridges can be formed, so I'm able to produce a little more strength. Now 8 cross bridges can be formed. I can produce a little more strength. Now all the cross bridges can be formed, and there's plenty of room to perform power strokes as well. So right now, we are able to generate our maximal tension. But there's one more thing we haven't drawn here. Remember, the other thing that is located here on our muscle cell is those 3 rows of myosin right at the M line, right, forming a smack dab in the center. Notice if we continue to shorten the sarcomere, there is still plenty of room for the myosin. All the myosin and actin are overlapping. All of the myosin heads can form cross bridges. But as the actin starts to interact with the myosin, is it going to be easier or harder for those myosin heads to perform power strokes? What do you think? If the actin's tangled up in the myosin, is it going to be easier to move it or harder to move it?
Harder.
Harder.
Harder to move it, absolutely. And if we go a little bit further, where the actin from the 2 sides is actually bumping into each other, it's going to suddenly be even harder. And if we shorten that as much as possible, even though all the myosin heads can grab, are any of them going to be able to form power strokes? No. And if none of the myosin heads can perform power strokes, what happens to our tension?
Sure.
There's no tension.
Decreases.
Yeah, it becomes 0, exactly. And again, if you don't like my art, we've got this great illustration from your textbook that does a really, really nice job of showing this. Right? Notice here, we see this relationship between the myosin and the actin. Notice the place where there is the highest tension that can be produced, the maximal amount of tension, 100% of the tension, is where all of the myosin and the actin are overlapping, but there is still plenty of space to perform power strokes. notice as we stretch it out more, as we elongate the muscle, less myosin can bind to the actin, and so we produce less tension. Conversely, if we shorten the muscle, the myosin heads can perform fewer power strokes. And if fewer power strokes occur, we produce less tension. So again, the key to this is how much tension we can produce is determined by how many myosin heads can bind, how many cross bridges we can form, and how many power strokes we can perform. So as we change the length of the muscle, we can change how much tension it can produce. And notice, most muscles, when we're in anatomical position, most of them are at or near their maximal length for maximal efficiency. Did you still have a question, Julia?
No, is my hand still up? I apologize.
No, I wasn't sure if I'd answered it or not. Okay, I put it down. All right, excellent. So, and again, think of back to the bar. If the bouncer isn't pulling the person's arms behind them, then they're wrapping them up in a bear hug. Because if your arms are squeezed down to you here, again, you're not going to be able to generate as much force. So if I can bear hug them and shorten their muscles, or I can pull their arms behind them and stretch them out, I can reduce the power of that individual I'm trying to subdue, and that makes it a lot easier to subdue them. Yes, question.
So this might be a little off topic, but when you're talking about like, or if you're like holding weight and you want to hold it close to your body, I understand for like, not like, so you don't pull out your back and stuff, but does like holding large amounts of weight closer to your body have anything to do with like muscle tension? Does that make sense?
It does make sense. I understand where you're going with that. No, because obviously it's related, because how much, it's going to be easier to lift up the weight or hold the weight at certain lengths of the muscles and maintain that contraction at certain lengths of the muscles. But a lot of time when you're holding those really heavy weights or carrying heavy boxes and doing things like that, the reason why form is so important is because it's about the physics of it. By maintaining your center of balance and by keeping it close, you're putting less stress on the muscles from multiple axes. So really, in that case, it has to do more with about the amount of axes. But having the right form, having your arms in the right position when you start to lift something, absolutely is going to be important, right? So getting that right positioning before you even start, right, has a lot to do with the arms behind you. If you're picking up a box, you don't reach way behind you to grab that box and pick it up. You keep it in front of you where you're able to grab it and much more easily lift it.
Okay.
Yep. All right. Questions on this? I always think this is the easier one, but we struggled a little bit with this one. So let's make sure this makes sense before we go on to the next one. All right, excellent. That's what I like to hear. That makes sense. Perfect. All right. The second factor that can determine how much tension a muscle produces, and we did all that, is how it is stimulated by the nervous system. Now, there is a new rule we need to learn. That new rule, and let's actually start this on the whiteboard. In general, which again, there's always things that can modify this, there's always things that can influence this. But in general, when you produce one neural action potential in a somatic motor neuron, you get 1 muscle action potential. Okay, so usually it's a 1-to-1 ratio in that fashion, and 1 muscle action potential in a skeletal muscle cell produces 1 twitch. All right, so basically what we have here, oops, nope, want this here. 1 neural action potential gives us 1 muscle action potential, gives us 1 twitch. So if we got that piece of knowledge in our pocket, then the next question is, what is a twitch? A twitch is 1 complete excitation, contraction, and relaxation of a single muscle cell. So we stimulate a muscle cell like my bicep brachia. It gets stimulated, it contracts, and it relaxes. Now, if I just send 1 neural action potential to produce 1 muscle action potential to produce 1 twitch, is that going to last for a tremendously long period of time?
No.
No, this can last somewhere between, I would say, about 20 milliseconds to maybe slightly over 100 milliseconds. We're talking about a very short event that is occurring. But regardless of whether it's very fast or very slow, there are, as the name seems to indicate, there are basically 3 main phases or periods to a twitch. Now, let's go back to the lecture for a moment. So again, it's a stimulus, excitation, contraction, relaxation of a single muscle cell. Remember, still at the level of muscle cell. And if we were to measure it, here we can see some examples. If you sent a single neural action potential to one of the muscles that moves your eye through space, you would get a single excitation, a single contraction, a single relaxation, and it would take a little less than 20 milliseconds. If you sent one to your gastrocnemius, and anyone know where your gastrocnemius is?
Is it your calf?
Yeah, it's the superficial calf muscle, absolutely. Notice that one would take about 40 milliseconds. And gastrocnemius was awesome. Anybody know where the soleus is?
In the foot?
Close. It's actually the deep calf muscle beneath the gastrocnemius. The gastrocnemius is the big superficial muscle on top. The soleus is right behind it. So your calf muscle is actually made up of those 2 things together. The soleus's job is, one of the things its job is to do is to basically keep you upright, standing. So it isn't necessarily a very strong or a very powerful or a very fast muscle, but you can stand for pretty much ever. So not surprisingly, a single twitch in your soleus lasts over 100 milliseconds. So these are some examples of these twitches, and notice whether they're really fast or whether they're really long, they all have the exact same type of stages to them. So you are correct. When you have a twitch, for instance, as you're falling to sleep, that would be an example of a single neural action potential producing a single muscle action potential producing a single twitch. Absolutely. But typically it's not all the cells. Your whole entire body doesn't twitch all at once, but you might just have one arm or a leg or something like that that twitches, right? It's the spontaneous release of electrical activity as your body is relaxing and calming down. That's the point of the twitch, is to release tension. All right, let's take a look. Let's pluck one of these out and look at the twitch. And again, even though this isn't something you're going to have to draw on an exam, it's still worthwhile to take a moment to draw it and see the components of it. Now, if you look back at that illustration, you may notice some interesting things. Now, of course, anytime we draw a graph, we need to label the components. So of course, here our x-axis is time, and our y-axis here is the amount of tension we are going to be producing. And if you look closely at that illustration, you'll notice something interesting. The first marker, which happens about yay, is what they call time zero. That is a little odd because normally when we think of an x-axis, zero is usually what's in the corner, right? But here it's a little offset. And in particular, the reason for this is what they mean by, and let's do that, and let's do that. This time zero is actually when we produce the muscle action potential. That's what the zero time indicates. It indicates when the muscle cell is stimulated. So why is this offset? What's happening in this time here, from here to here? Any idea what's going on right here before the twitch begins?
Would it be like a resting? Nothing.
Well, the muscle is resting, absolutely. But think of it this way. When you decide to contract a muscle, when I want my muscle, my bicep brachia to contract, does it instantly produce a muscle action potential the absolute instant that I think it should contract? Does it suddenly start producing a muscle action potential right away?
No.
You have to wait for it to get to your muscle, I guess.
Right. We have to make the decision. We have to spread that neural action potential to the synaptic end bulb. We need to open those voltage-gated calcium channels, right? Absolutely. Communication time, exactly. If you think about it, what's happening right here before the zero time are all the events of the communication at the neuromuscular junction. Right? You are absolutely correct. That is everything that is happening in this zero time. This is everything before the muscle actually gets that muscle action potential. So basically, it's the first part of this, right? From the decision, the neural action potential, to our muscle action potential, right? When we get our muscle action potential, that's what gives us our twitch. And that's what we see next. So that muscle action potential, which again, wasn't the zero, is the zero time because that's when it's excited, but you're absolutely right. We first have to make the decision. We have to send the signal, release the acetylcholine, have it spread across the synaptic cleft. All those other things have to happen first. But once we get the muscle action potential, we produce our twitch. And as I mentioned, our twitch has 3 phases. Oops. If we were to draw this, and we'll take the gastrocnemius one because it was at 40 milliseconds, and that's as good a starting point as any. So let's draw a line. Let's pick a color. We'll do black for this first one. No, actually, I don't want to do black. Let's do red. Ooh, let's actually make this bigger. There you go. It's not a very symmetrical, but we get the idea. This is our twitch. And as I mentioned, our twitch is going to have 3 phases. And we can see those 3 phases as they're indicated by the change in tension. Phase 1 is what is known as the latent phase or the latent period. And when you look at the very beginning of our twitch, how is our tension changing? Is it going up or is it going down?
It's staying neutral.
Staying the same, exactly. Notice during our latent period, there is actually no change in tension. The tension stays 0. Why not? What's happening? Let's do it this way. Why isn't there any change? When we get the muscle action potential, don't we instantly start producing tension? Isn't it instantly generating force?
No, because
No.
it has to spread down the sarcolemma and do all that.
Yeah, absolutely. Exactly. Do all that. I like that. You've absolutely got the right idea. Our muscle action potential spreads. As it spreads, it opens the voltage gated calcium channels. Our calcium has to come out into the cytosol, right? It needs to bind to troponin, right, etc. All of those things. And was there a term we used collectively for all of that?
The excitation,
Yeah. Excitation, contraction coupling.
action coupling.
Absolutely. Before we can start generating tension, excitation, contraction coupling occurs. So notice during this, during this latent phase, that's the one nice thing about being a bad artist and also having this on the board is I can actually move my lines, which is awesome. During this first stage, stage 1, the latent period, there's no change in tension. And that's because, remember, what causes the change in tension is myosin heads grabbing onto the actin and pulling. And while the muscle action potential is spreading, while we're opening those voltage-gated calcium channels, while the calcium is coming down to the cytosol, again, those things don't take a lot of time. This latent period only lasts about 1 to 5 milliseconds. It is a relatively fast process. Right? Heck, 1 to 3 milliseconds or something like that. It is a relatively super fast process. But there is a little bit of time while those things are occurring. And during that latent period, there's no change in tension. And that brings us to phase 2. Phase 2 is our contractile phase, or the contractile period. And how does tension change during the contractile period? Well, what happens? Tension increases, exactly. So during the contractile period or the contractile phase, our tension is going to increase. Why? What causes the tension to increase? There we go. Arrow's got it. Right? We get tension because myosin heads grab onto the actin and pull. Now notice it doesn't instantly go to its maximal tension. Why not? Can anyone figure out why not?
What we talked about earlier, how not all of them are pulling.
True, some of it could be the length of the muscle determining that it's not able to necessarily pull its most efficient tension at first. But let's think of it this way. When those voltage-gated calcium channels open, does all of the calcium all come out at once?
No.
No. So notice what's going to happen is we are going to get calcium starting to come out. So calcium levels increase in the cytosol. And as they increase in cytosol, we're able to move more regulatory proteins. And as we move more regulatory proteins, more myosin heads can grab and pull. And so we get an increase in tension. The tension is going to go up. So we get an increase in tension here. Tension is going up during phase 2. because more calcium is being released. And as more calcium is being released, more regulatory proteins get moved, and more myosin heads can grab and pull. All right. We comfortable with that? Now, if I'm just sending one neural action potential and I'm just producing one muscle action potential, that one muscle action potential is a big, huge positive signal. But as we've talked about, does it stay around for a long period of time?
No.
No. So just as quickly as the cell depolarizes, opening those voltage-gated calcium channels, letting the calcium come out, that cell's going to repolarize just as quickly, closing those voltage-gated calcium channels and not letting any more calcium come out. This is what we call the relaxation phase, or the relaxation period. And based on what we see here in the illustration, what happens to tension during the relaxation period?
Decreases.
It decreases. Notice relaxation is not when it's at 0. When it's at 0, and hold on, I need to cheat, move this a little bit out of the way. Grab this a little bit to move it that way. When it's back at 0, and I guess I'll have to use an arrow to do this. This period here, it's back at rest. 0 tension is at rest. Let's actually write that. That's not what we're talking about. We're talking about the relaxation phase. And during the relaxation phase, tension decreases. And guess why tension decreases? It'll make cells do wonky things.
You have a decrease in calcium.
Exactly. Notice, once we close those voltage-gated calcium channels, no new calcium enters into the cytosol. But remember, we still have a lot of calcium in the cytosol. And does that calcium in the cytosol instantly, all of it get put away?
No, it gradually decreases.
Yeah, it's going to take some time, and that's exactly what happens. So calcium levels decrease in the cytosol. And if there's less calcium, that means less regulatory proteins get moved. And if less regulatory proteins get moved, less myosin heads can grab and pull. And if less myosin heads can grab and pull on the actin, then less tension is produced. And in that fashion, our tension will ultimately go back down to 0. And that is a single twitch. Now again, most, if not all of you, have experienced a twitch before, right? Twitch isn't the main way I lift up this Coke to my mouth to take a drink, right? If I tried to do that with a bunch of twitches, it would be all over the place. It would be a mess. So clearly it's not the main way I'm going to move my body through space, but it is the starting point for how I'm going to get a smooth movement through my space, through my movement, through my hand, through my arm, lifting it up. This twitch is our starting point. Because ultimately, when I want to move my body through space, I don't have to rely on just one neural action potential. I can produce many neural action potentials, which are going to produce many muscle action potentials, which will produce many twitches that I can put together to move my body through space. But this is where we have to start. We have to start with a single twitch. And again, if you don't like my pictures for this, let's look at the pretty pictures from your textbook. Again, here we see our latent period. Let's use blue because they haven't. Latent period is this first part. Like I said, 1 to 3 milliseconds. The shiver is typically something different. So, I mean, again, any muscle movement is going to involve twitches, but it isn't just an individual twitch that you're feeling as a result of there. And also, a lot of that shiver also has to do with the erector pili muscles as well, and your skin and other factors like that that are going in. So, again, any movement of any muscle starts with a twitch, but we're not just seeing one twitch that's causing it. All right, latent period. Zero change in tension. And what did we say is occurring during this latent period?
Excitation, contraction, coupling.
Excellent, perfect, all right? Contraction period. How does the tension change during the contraction period?
It increases.
It increases. Why?
Because the calcium levels increase in the cytosol.
Excellent.
And then they move more regulatory proteins.
Perfect. More calcium in the cytosol, move more regulatory proteins, more myosin heads grab and pull. And then we start locking up the calcium. Less calcium is present, fewer regulatory proteins move, fewer myosin heads can grab and pull. So what happens to the tension there?
Decreases.
Decreases. There you go. And that is our relaxation period. And that is a single twitch, end of story. Questions on that? Okay, stunned silence means, yes, Dr. Slutsky, we understand this tremendously. Make the exam harder. So excellent, that's what I love to hear. So with that in mind, if we're completely comfortable with a single twitch, then let's add one more component to it. As I already mentioned, if I'm going to move my body through space, am I going to rely on just one twitch? No. So as we've talked about, while this is great to understand what's going on with one twitch, we need many twitches to move our body through space. And there's one minor problem with this. That sounds like a great idea. Excellent. Let's just pile on the twitches so we can move our body through space. But there is something very, very interesting about our muscle cells. Remember, as we just finished talking about, one neural action potential equals one muscle action potential, which equals one twitch, right? We did that? Make sense? But what happens with our muscle cell, when our muscle cell produces a muscle action potential, there is a brief period of time. And I do mean brief, we're talking 1 to 5 milliseconds. But there is a very brief period of time where it loses its irritability. Remember, irritable means that it is able to receive a stimulus and respond to it. It gets the neural action potential, it produces the muscle action potential. Well, during this very brief period of time, it loses its irritability. What that means is it cannot produce a second muscle action potential. And if you can't get a second muscle action potential, guess what that means? Well, what does the muscle action potential give us?
It
Yeah, well, it gives
stops.
a twitch, right? Well, again, if we can't produce a second muscle action potential, then we can't get a second twitch, right? So if it loses that irritability, if we can't get a second muscle action potential, we can't get a second twitch. This brief period of time is what we call the refractory period. All right. Yeah, we've all experienced a refractory period at one point in time or another. You finally lure that cute young man home, get what you need out of him the first time, and then you can't get it out of him the second time again. He has some difficulty rising to the occasion right away, right? You got to give him that little bit of refractory period to recover before you can get the rest of what you need out of him, right? It loses that ability to respond. It loses that irritability. Now, notice we're talking about 5 milliseconds. That's from when we produce the stimulus the first time to this point here. Notice we've barely started the twitch at this point. But even though it is a very small period of time, this period of time, this refractory period, is the period where we cannot produce a second twitch. Notice it has nothing to do with the first twitch and everything to do with when we can produce a second one. Socially awkward or socially prepared? Exactly. I'm telling you, you walk up to some young man at a bar and tell him, hey, did you know the calcium levels in your blood need to be between 8.5 and 11 milligrams per deciliter? And he will just swoon when you say that to him, right? A and P is the language of love. There you go. All right. So this refractory period, go ahead and erase all that. Again, has nothing to do with this twitch and everything involving our ability to produce a second twitch. That's a great way to do it, right? Grab some washable markers, right? And then just write on your kid, write on your significant other, write on some random kid from your neighborhood, write on their arms and legs, identify the muscles and bones. It's a great way to learn this material. All righty, questions on this? Again, if this single twitch doesn't make sense, it's just going to get worse from here because everything we're adding from here adds to what we already know. So I'll ask one more time. Any questions on the twitch before we move forward?
Not yet.
Arthur, what I'd encourage you to do next time you order something through Uber Eats, just when the guy gets there, just ask the driver when he gets there if you'd mind if you wrote on him for a little bit to help study for an exam. I think that's probably the best way to do it. All right. All right, excellent. Exactly. All right, perfect. So if we understand a single twitch, then let's take this to the next level. Because as we said, a twitch is useful, it is vitally important, but it's not the way we're going to move our body through space. So let's redraw our graph here for a second. I need to move that over here, get it out of my way. Move this over here. All right. All right. So we're going to drop our scale a little bit, but you guys will get the same idea here. And we'll use red. We know that if we generate a muscle action potential, that muscle action potential will produce a single twitch with a latent period, an a contractile phase, and a relaxation phase. Now, what would happen if, so I had my Petri dish, I had my muscle cell, and I got a twitch, and then I waited 5 minutes, and at 5 minutes, I stimulated it a second time with a second muscle action potential, generated a second muscle action potential. What would happen?
It would overlap.
Well, 5 minutes later, is that first twitch going to last 5 minutes?
That would be the
No.
No, but you're right. I'm going to produce a twitch. And how would this twitch relate to the original one?
It overlap.
No, well, by overlap, I think I was getting the right. They'd be the exact same size, right? They would both produce the exact same amount of tension. Now, again, I know my drawing's a little off on that. I think I can cheat and maybe stretch that out. There, I think that looks a little better. Are those close? Let's pretend those are the same.
So it would never occur to where the second one would be larger.
Well, we're getting there. Hold on. So again, I'm cheating. So let's pretend that that's the same. Now, again, remember, I've waited 5 minutes. So I had the first one that happened at time 0, and then I waited 5 minutes and stimulated it again. And when I stimulated it again 5 minutes later, I get the exact same twitch from the exact same muscle, from the exact same neural action potential, from the exact same muscle action potential. I get the exact same twitch. However, you have the right idea. I don't have to wait 5 minutes to do it again. So what happens if I wait till, again, notice the tension equals 0. I let all the tension, that does not need to be in caps or this big. And as soon as the tension reaches 0, I stimulate it a second time. So here I'm going to produce a second muscle action potential. And what's going to happen with this second muscle action potential? What am I going to get from a muscle action potential? What do muscle action potentials produce?
A twitch?
A twitch?
A twitch, absolutely. And how do you think this twitch is going to relate to the first one? Is it going to be identical again?
No.
Maybe be smaller.
Now, in this case, what actually happens is it's slightly bigger. Now, if I then wait 5 minutes and stimulate it again, it's going to be back to its original shape. But notice what's happened here. I stimulate this one a little bit bigger. A muscle cell immediately after it reaches 0 tension. And as a result of that, I produce a slightly stronger twitch. This characteristic of a muscle cell is the characteristic we call TREPI. Skeletal muscle cells have this characteristic called TREPI, where if we stimulate a muscle cell immediately after it reaches 0 tension, it produces a slightly stronger twitch. And what's cool about this is it's repeatable. If I again let it reach 0, and when it reaches 0, I stimulate it a third time, I'm going to get a third twitch, and this third twitch is going to be bigger than the next one. And then I stimulate it again, and I get an even bigger twitch. Okay? In fact, in some muscle cells, this can be repeated as many as somewhere around 30 times. And each time it gets bigger. Now, eventually, it will reach a maximum. Maximum TREPI. So whatever that maximum TREPI is let's say for argument's sake that this last one was, well, or let's say this next one is going to be the maximal TREPI. At this point, every subsequent trepi is going to reach that size and then just come back down. So it'll never go higher than that. There is some maximal trepi, but it may take 30 times to get there. Now, notice and I'll put this in capitals because I want to emphasize it. We let the cell reach zero tension, right? This is very important because notice what this means is we are not adding twitches together. It's not like one twitch already has some tension and we're adding to it. But we are getting a bigger signal. And so my question for you is, why? What could possibly be happening here that allows us to get a bigger, stronger contraction after it reaches zero tension?
Would it
I'm
be
guessing
the buildup of calcium being released?
What were you going to say? Who else? Sydney, what were you going to say?
Oh, I said the buildup of calcium being released.
All right, who else was talking? Someone else was
I
talking.
was gonna guess the same thing. It's like the calcium's already kind of built up in your cell.
Absolutely. You guys absolutely hit it on the head. What's happening here is residual calcium. Oops. Right? Let's think about this in really simple, really artificial numbers. Let's say, for argument's sake, that when we produce a muscle action potential, 100 calcium are released. And that's what produces the twitch. When the cell gets back to zero tension, is every single one of those zero calcium going to be put away?
No.
No, maybe only 90 of it is put away. So there may be 10 left. So when we stimulate it a second time, we know that stimulation releases 100 calcium, but there's already 10 in here. So now we have 110 total. More calcium, move more regulatory proteins, and as a result of that, we get more tension. Again, 90 get put away, so now there's 20 left. And then we stimulate it again, and now there's 100 that are released, and now we have 120 total. And again, these numbers are completely made up. They're totally fake, but you're getting the idea. What's happening is there is residual calcium left over, even though the cell reaches zero tension. So let's start with that. The cell reaches zero tension, but there is still residual calcium left over. And that residual calcium left over, when stimulated immediately again, is able to produce a stronger contraction. Now, I'm fairly certain that almost none of you have thought about residual calcium before and this concept of trepi. But I also know that every single one of you is aware of this. Because right around the corner, we are going to have the Summer Olympics in Tokyo for 2020 here in 2021. And one of the best, most amazing, fun events to watch is the 100-yard dash. All right. Now, if you think of those sprinters as they are moving across the field to get into their blocks to start that race, we all know that they move as slowly and as quietly and minimize their movements as much as possible. They try to stand still as much as possible so they can conserve all of their energy to be able to burst out of their blocks. And is that what those sprinters do as they're getting ready into the blocks? Are they standing as still as possible?
No, they're jumping around.
No.
No, they are in constant movement. They are constantly moving, constantly jumping around, constantly moving their legs and their arms and wiggling as they're getting into it. They don't hold still until the very last second before they take off. And why are they in constant movement? Because they want to be releasing this calcium. They want to have lots of excess calcium, residual calcium in their muscle, so that when that gun goes off, they're producing a maximal tension contraction so they can burst out of those blocks. Right, so that
Yes.
constant movement, that constant moving around, right, helps the stretching before you exercise is important for the joints, but it's also important for the muscles. Right, so that again, if you're getting ready to do deadlifts or bench press, you don't start at the maximal rate. You work your way up to it. You warm up to it. That warming up of the muscles helps to not just help the joints by increasing the fluid in the joints and protecting the joints like we talked about in the last section, but it also warms up the muscles and gets the muscles to be more efficient as well. All right, so again, you probably have never thought of Trepi before, never thought of residual calcium before, but all of you have seen this in action. And all of you, if you didn't fully understand why they were doing it, you knew that they were constantly moving before they got in. They weren't conserving energy, right? They were moving because they were trying to get that calcium so that they can produce that powerful contraction as they come out of the blocks. Trepi means staircase? That's excellent. I like that. I didn't know that, but that's great. Yep, and these absolutely work in a, oh, cool. Absolutely work in a staircase fashion. Excellent. Again, I, with my limited drawings, have done this, but again, we've got the pretty pictures from the textbook. So again, when we talk about this concept of Trepi, again, the key to this is we have a second stimulus immediately after the relaxation phase. That second stimulus enhances the first, and we get slightly larger contractions, right? Till we reach that maximal tension in Trepi. And like I said, 30 up to 50 times to reach that maximum. So those sprinters wiggle, right? Constant movement, getting ready, warming up those muscles, trying to get them as efficient as possible. But this still doesn't take me to my drinking problem, because when I'm trying to lift that Coke up, right, it's not like I take a weak twitch and then a weaker, slightly stronger twitch and then a slightly stronger twitch and then a slightly stronger twitch until finally the twitch is strong enough to get the drink to my mouth. All right, that's not how we do it. So we're still not quite to the point where we're moving the body through space. But here's the deal. Do we actually have to wait for the cell to reach zero tension before we stimulate it a second time?
No.
No. And so the way we typically move our body through space is a process we call wave summation. In wave summation, as the name indicates, we are adding waves of tension together. Right? Wave summation is just like it sounds. We're adding waves of tension, or you can think of it as twitches. We're adding twitches together. Now, obviously, to add these twitches together, there have to be two rules. We have to stimulate it the second time before the tension reaches zero, because after all, if you add something to zero, you're not adding it to anything. So obviously we have to do it before the tension reaches zero. However, remember, we can't do it instantly because there was that very brief period of time where our muscle cell loses its irritability, where we can't produce a second twitch. And what did we call that brief period of time?
The latent period?
Or refractory period, absolutely. So notice it also has to be after the refractory period. But remember, that refractory period is only about 1 to 3 milliseconds. No 1 to 5. Let's say it that way. So before it reaches zero tension, but after that refractory period. Let's go back to our illustration. Notice with this first twitch, and I will switch colors down to something bright and colorful. We'll add pink. All right. Anytime from after the refractory period, remember the refractory period occurs in that first 5 milliseconds, but before it reaches zero, if I stimulate it and produce a muscle action potential, then what's going to happen is I'm going to get a second twitch with a latent period, a contractile phase, and a relaxation phase. And if I stimulate it again before it reaches zero, I'm going to get a latent period, a contractile phase, and a relaxation phase. And stimulate it again, and stimulate it again. And in this fashion, I can keep adding twitches together. And when I keep adding twitches together, I can produce much tension. As a result of this.
And this all happens fairly quickly, right? Like within a couple seconds, you can go through.
Even we're talking fractions of seconds here. We're talking about hundreds of milliseconds. Absolutely. All right. Now, again, is there going to be some peak? Is there going to be some maximal tension we can produce with wave summation? What do you think? Yes or no? Or can our muscles just produce infinite amounts of power?
There will be a peak for sure.
Yeah, absolutely. There is going to be a peak. And what's interesting is under normal function, we do not reach that peak. In the normal function of our muscle, typically what will happen is we will get to a point where we get this slightly undulating, oops, I ran out of room on my mouse pad. This undulating tension of peaks and troughs, undulating tension of pressure, undulating amounts of tension near the peak. This is what we call an unfused tetanus. All right. With this unfused tetanus, this is how we typically move the body through space. Now, this seems kind of weird to me because, again, when I'm trying to lift this Coke to my mouth, is it shaking all over the place from these constant changes in tension in the muscle cell?
No.
No. So why is that? Why isn't my hand all over the place while I'm trying to lift that soda to my mouth?
It's not it's like a smooth contraction, not a twitch, if that makes sense.
Well, but remember, right now we are talking at the level of a single muscle cell. Now, let's say that I decided, because I was so inclined, that because I know you guys struggle on the exams, I decided to give extra credit. And for extra credit, all you have to do is stand up in front of the entire school and sing a solo. And if you do that, you will get 30 points of extra credit.
What song is it?
I don't know. I get to choose. Some aria probably or something like that. How many of you would be willing to stand in front of the entire school? A couple of you would. Okay.
Yes.
Really?
Give me the location, the time, the address, everything. I'm ready.
All right. Well, so some of you are more bold than others, but I'm guessing there are lots who necessarily wouldn't. No, you got to do it live in front. All right. However,
the vlog.
Okay. But how many of you would be willing to do it as part of a 100-person choir?
That's fine. I'm gonna vlog too.
Yeah, exactly. Well, I don't think there's anybody who, so while most of you seem willing to sing in front of the entire school, and again, there's over 10,000 people in our school, so you guys are much more bold than most of my classes usually are. But the point is, it's easier in a choir because you don't stand out as much. And that's what's going on when I'm lifting that drink to my mouth. Yes, that soda is relatively small, but it's still, I'm not just using one muscle cell to lift it. I'm using many muscle cells to lift it. And when you have many cells working together, even though each individual one is undulating, together, like a choir, they smooth out to be a much more uniform movement. All right? However, if you're trying to do an activity that only uses a few muscles, like for instance, trying to thread that needle, right? When you're trying to thread that needle, that becomes a little bit more challenging because when you're doing a very small dexterous activity like that, you're only using a couple muscle cells. And when you're only using a couple muscle cells, you only have that, that trio or that duet, right? Then the individual muscle cells and their actions stand out and it's much more obvious. So when you're lifting something up normally, you are much more uniform in that action because they're all going to be working at different rates, and so it all smooths out. At the same time, if you are using all the muscle cells together, like you're trying to help a friend move a couch up 3 flights of stairs, right? After a while, as you're holding the couch, because all the muscles are firing together, your muscles might start to shake because of that, because again, you're seeing this unfused tetanus, because instead of firing asynchronously, they're all firing together because you're near your maximal weight that you're trying to lift. Now, does that mean that it is completely impossible to reach that maximal and stay there with a nice, smooth, sustained contraction? Yeah, bodybuilders shake when they're lifting those heavy weights. Absolutely. That's a great example of it. Is it possible to get a muscle to contract at its maximal strength and that nice, smooth, sustained contraction? What we would actually call a fused or complete tetanus. It is possible, but it's not healthy for the muscle in most cases because you're producing so much tension that it could be potentially damaging for the muscle cell. So it's better to use multiple muscle cells together to do that. But certainly artificially, if you grab onto that electrified fence and you have an artificial signal going into all the muscles, all of those muscles are going to all contract at their maximal. But other muscles can be trained to get better at this as well. When you are performing surgery, when I was in graduate school, one of the things that my PI, the primary investigator that I worked with, had us do is you had a mason jar with a dice at the bottom of it, a single die at the bottom of it. And using suture and 2 forceps, you had to tie a bow around that die inside of the jar. And the reason you do this is because A, they like to punish you and torture you, and it's hard and challenging, but you get used to using those muscles and helping to smooth those muscles out. So, because again, you don't want your surgeon's hands shaking as they are trying to do your brain surgery or something like that. So again, it can be trained. You also hear stories about little old ladies lifting lifting cars off of babies and stuff like that. So, again, it is possible to get it, but it's not typically the way we move our body through space. So you have the right idea. So someone was asking again about that electric shock. Yes, the problem when you grab onto that electric fence is that electrical signal stimulates all the muscles, causing all the muscles to produce a muscle action potential. And all the muscles are producing action potentials at the same time. Right? So you can't relax the muscles because all the muscles are contracting. If all the extensors and contractors and flexors are all contracting at the same time, you're locked in that position. And that's one of the damaging things about being electrified is that you can't let go. What is the grape surgery? I don't know what a grape surgery is. To do what? I never did surgery on a grape. I don't know what a grape. Just so that you're delicate at it. I don't. You have to peel it or something. Is that what they do? Oh, okay. I had not heard of that. That's cool. Yep. No, I didn't have to do that in mine. I had to do the dye thing, but I didn't have to do. I never had to. I never had to do anything to a grape. All right. My wife makes me peel the grapes before I feed them to her, but I never had to do that for a medical procedure. All right. Excellent. So I've done an amazing job of drawing this, but let's again come back here and talk about this. Here is that concept of wave summation where we're adding those waves together. And as I mentioned, the primary way we move our body through space is with an incomplete, or what we call an unfused tetanus, where we are producing undulating rapid contractions and relaxations near the peak tension for the muscle. But it is also possible, theoretically, to produce a complete tetanus, a smooth, sustained contraction at the peak tension level. And that is how we change the tension in a single muscle cell, by how rapidly we contract it, the length of the starting point, and how rapidly we stimulate it, I mean. All right. And so with that, we are done with the lecture for today. That is excellent. So like I said, we didn't have as much today because we're behind on the lab. So I want to catch up on the lab. So this is a good stopping point. That is our game plan. And we're now done with a single muscle cell. So now we're going to start building up into groups of muscle cells as we continue to work our way upward and onward. Excellent. So what we have left to do for the rest of today is to finish up, or at least try to catch up. I don't think we're going to fully catch up today, but we're going to try to catch up, do as much as we can with the origins, insertions, and actions. So towards that goal, let's go ahead and take one last break. Get your red pens and blue pens and handouts. And let's meet back here at 2:33. And we will restart with the lab at that point, and I will start the recording at that point as well. Excellent. All right. So any questions before we switch gears into lab mode? All right. Excellent. I will see you guys in 15 minutes then. All righty. Let's go ahead and get started. So we left off last time and we were still working our way through the muscles of the trunk. We had identified all of the muscles that we needed to know the origins and insertions of. And so we'd moved on to the ones that we don't need to know the origins and the insertions of. And we had done the two diaphragms, the actual diaphragm and the levator ani, which is part of the urogenital diaphragm. That leaves us with a couple of groups of muscles. And this first group of muscles I want to identify are the intercostal muscles. As the name indicates, where are the intercostal muscles found?
Costals.
I'm sorry, say again?
Between
Intercostal means what?
in between the ribs.
In between the ribs, absolutely. So obviously, for that, we need a sternum. So let's go ahead. And again, I'll spend a tremendous amount of time and effort drawing my sternum as carefully as I can. There you go. That's very clearly a very obvious sternum. And let's draw some ribs. There's rib 1. There's rib 2. There's rib 1. There's rib 2. You can see the tremendous time and effort I'm putting into this. And because we can't forget it, the ribs we know, because we'll assume that these up here are some true ribs, we know they're going to directly connect to the sternum via cartilage. So actually, I don't like that. Let's use orange. Is that more noticeable? Perfect. All right, excellent. So again, super simple, super quick, but it will serve our purposes really, really conveniently. There are two types of intercostal muscles. They are the external intercostals and of course, then what must the other one be?
Internal.
Internal intercostals. Now, based on that, what do you think these two names, what do you think the most obvious difference between these two are?
One of
Their
them.
actions.
True,
Inside,
their actions are, but even more obvious than that, based on the names alone.
location is different.
And what do you mean by location in this case?
So the internal would be facing like the heart and lungs, interiorly, medially, all that jazz. And external would be on the outside of ribs.
Excellent. And so do we have special anatomical terms for closer to the outside and closer to the inside?
Deep and superficial.
Deep and superficial. Excellent. So which would the internal intercostals be? Would those be deep or would those be superficial?
Deep.
Excellent. So these are going to be the deep. And our external intercostals are going to be superficial. But so super obvious in their differences anatomically that way. But there are two other big important anatomical differences. Our external intercostals are also more lateral. And the other big thing with our external intercostals is that their fascicles project down and in. So if we were to draw these, they start more lateral and their fascicles project down and in. Now, not only do they start more lateral, but they don't go all the way to the cartilage. They don't go all the way or touch the sternum. And again, their fascicles come down and in on both sides. So they are superficial, they are more lateral, and their fascicles go down and in. Our internal intercostals are deep. They are more medial. They actually are going to be in contact with the sternum, in contact with the cartilage of the ribs, and their fascicles project down and out. So if we were to draw them, they would start here medially from the sternum, from the ribs. They would project down and out. And remember, because they're deeper, they would start to get covered up by the external intercostals. So again, they're projecting down and out. And again, they are going to be deep to the external intercostals. So when we look at these things anatomically, and notice your book does a great job of showing this. Notice when we look at our illustration here of them all individual, we can clearly see here are the deeper muscles, and they are more medial, and their fascicles are going down and out, where here more lateral, here more superficial, and their fascicles are coming down and in. So we can see the externals and we can see the internals in between the ribs. And what's really cool is they actually have this great illustration where they've actually peeled apart the two muscles. Now, clearly, when you've peeled apart the two muscles, we can't tell which one is superficial and which one is deep, but we can clearly see how the external intercostals are more lateral. Notice they don't come to the sternum, they don't come to the cartilage. And again, their fascicles are coming down and in, whereas our internals clearly start at the sternum, clearly attached to the cartilage, and their fascicles are going down and out. So anatomically, these are very easy to identify and distinguish. All right, questions on the anatomy of them. Notice we don't need to know the origins and insertions, but we know they're between the ribs, and we can very clearly tell them by their structures, by their orientation, by their anatomy, by their location. But we also need to know their actions. And these are what we call accessory respiratory muscles. Their job is to help us to breathe. Remember, as we talked about, while you're sitting here calmly in class, just sitting here and listening, you're primarily just using your diaphragm. All right. So again, if you look over at your loved one who's half asleep on the couch while you're going through lecture, you don't see their chest heaving as they're sitting there taking a resting breath. However, if you need to forcibly move air out, like I am while I am speaking, right, or you're exercising or something like that, then you need to be able to have other muscles that help in this. And we talked about how the sternocleidomastoid, the scalenes, the pectoralis minor, all of those are accessory inhalation muscles, and something like the something like the serratus anterior can help to force air out. So notice we have two respiratory processes, I inhalation and E exhalation. And here we have an I internal intercostal and an E external intercostal. So based on all of that, and of course, what is it that the internal intercostal does?
They,
If you're inhaling.
It would be really convenient if the internal intercostal inhaled, but is that what it actually does?
No.
No. It would certainly be convenient that way, but it ain't what it does. Our internal exhales and our external inhales. So it's not quite as convenient, but as long as you can remember that. Our internal intercostals basically bring the ribs down for that forced exhalation, and the external intercostals bring the ribs up for that inhalation. So while I-E-E would be convenient, it's also just as easy to remember that they're the opposite. I internal, E exhales, and E external, I inhales. So there you go. We have the muscles and we have the actions. Questions on those? All right, excellent. I wanted to make a point of doing the diaphragms first because I want to talk about the external intercostal and the internal intercostal, because when we get to our abdominal girdle, there we have an external oblique and an internal oblique. And there are definitely going to be some similarities between these two sets of muscles. So let's do that. Let's go to our next big muscle group. And our next big muscle group is the abdominal girdle. What is the function of the abdominal girdle?
To hold in the contents of the abdomen?
Yeah, absolutely. Provide protection, protect the organs, right? We have bones up here that protect our heart and our lungs. Do we have bones down here protecting our small intestine and our large intestine?
No.
No, and yet they don't go tumbling out of our body. I think I told this story in the last class. That's how Houdini died, right? And then when he was unprepared, he got punched in the stomach and his spleen ruptured, right? Because he wasn't able to engage his abdominal girdle. So all we have protecting most of the organs of our abdominal pelvic cavity is a muscular wall. So to make sure it is as protective as possible, we are going to have these at lots of different orientations. So lots of different muscle layers at lots of different orientations are going to provide multiple layers of protection. However, remember, we always have to worry about what bones it is going to be related to. And in this case, our abdominal girdle is primarily going to be involved in affecting the movements of our vertebral column. So let's put some vertebrae here along the back wall. So here are our vertebrae. So we can be thinking about how our different muscle groups are going to affect the vertebrae with their actions. So let's start simple. One of those simple muscles, and let's do that, and let's do that, and let's do this. We have some muscles that are long, straight muscles that basically run parallel to the vertebral column. They run vertically up and down the length of our abdominal pelvic cavity. And like most muscles, they're paired and found on both sides of the body. And what are these long, straight muscles in the abdominal region called?
Transverse Abdominis? Or
Transverse is a good guess, but what's the other straight ones? The vertical ones.
Rectus Abdominis?
Rectus Abdominis, absolutely. Rectus Abdominis are these two straight lines of muscles. And again, actually, that's what rectus means. Rectus means straight. This is the straight muscle in the abdominal region. One of the interesting things that I know you're aware of with the abdominal muscles, these rectus abdominis muscles, is there are tendinous insertions on both sides of these that divide our rectus abdominis into not one, not two, not six. There you go. Not one, not two, not six, but an actual 8-pack of individual bellies. All right, so that 8-pack of muscles is the rectus abdominis muscles. The way you make them visible is by removing all of the fat and dehydrating the skin above them. It's just that simple. What's interesting about these what's interesting about this rectus abdominis muscles is each one of these individual bellies is actually individually innervated. So it is possible to just contract one of those 8-pack bellies. Now, again, just because it's capable doesn't mean that you're consciously able to. All of us have an elevator muscle for our ears, but only some people are actually able to voluntarily control it where they can wiggle their ears in space. Anybody in here able to wiggle their ears and demonstrate it for the class? No one willing to admit it? You can wiggle your ears? Will you demonstrate it for us?
Sure. Let me see if my camera's working.
Okay, you're on.
Yeah, my dad taught me. Do you guys see me? Because I don't see myself.
We see you.
My dad taught me.
There you go. Awesome. That's spectacular. Excellent. Absolutely. See, we all have the muscles there to control it, but being able to do it isn't something that everybody is able to voluntarily control. That is spectacular. And it's the same thing with these. Everybody has this innervation, but not everybody is able to manipulate it that way. Terry Crews is a great example. There is a That's awesome. There is a video where he has electrodes connected to the different muscle groups, and by tensing the individual muscles, he can produce an electrical current that plays a musical instrument. So he has all these drums and other musical instruments he can play just by contracting the muscles of his body. So it's a great YouTube video, something that you can see. And so, yeah, so you have those are the rectus abdominis. Now, the question becomes, these basically run parallel to the vertebral column. So when we think of the actions of these, is this going to be able to twist or turn our vertebral column at all? Yeah, TED Talk, that'd be awesome.
No.
No, so notice it's not twisting or turning the vertebral column. So how many actions do we have for the rectus abdominis muscles?
2.
2. What are they?
Flexing the vertebral column and compressing the abdomen.
Excellent. The abdominal girdle as a whole, basically, as we've talked about, form a circular muscle group around the abdominal cavity. And so when you tighten these muscles, it compresses the abdomen, right? There you are on the beach hanging out for spring break. And it turns out the Jonas Brothers are recording their new video. So what, of course, do you do? Right? You suck in the belly so that they will notice. That compression of the abdomen. That's what we're talking about there, right? And then, of course, also that flexing of the vertebral column as well, right? That anterior flex. So there you go. Flexion of the vertebral column and compression of the abdomen. Now, as someone mentioned, we also have a deep muscle layer. And that deep muscle layer is basically perpendicular to this one, where the muscle fibers basically run horizontally. And this horizontal muscle group, what's this one?
Is it the obliques?
Well, remember, oblique means angle, though, right? This one, notice, is perpendicular to the rectus. This one almost goes across the abdominal region, almost like a transverse section. Absolutely. This is the Transversus Abdominis. And this is our deepest lateral layer. So it is deep, running, horizontal. And notice, again, if you think about the way these fibers work, none of them actually cross from one vertebrae to another. They basically just come out from the side and then come around towards the front. So once again, essentially all these is a circular muscle. So as a circular muscle, right, they don't have any ability, any ability to affect the vertebral column. So what is the one action that the Transversus Abdominis have?
Compression.
Right, compression, absolutely. Compression of the abdomen, making that abdominal cavity smaller. Now, as was mentioned before, we also have 2 more. We have an external oblique, angled muscle, and we have an internal. Now, just like we saw in the intercostals, what's the most obvious way these 2 are anatomically relative to each other?
One's deep and one's superficial.
And of course, which one's the deep one?
Internal.
Internal, just like the intercostals. And just like the intercostals, the external oblique is superficial. Remember, so we'll talk about that in a second. Let's finish talking about this and then we can talk about hernias. Absolutely, we can get to that. So that is the way they're different. Remember, another similarity between the external obliques and the external intercostals is in both, again, the fascicles project down and in. And again, in the internal oblique, the fascicles project down and out. So that's going to be the same that way. Remember, in the intercostals, the internals are more medial and the external are more lateral. We have something similar here as well, but in this case, the internals are more inferior. They actually do not, they let's say it this way, come up only to the ribs. Whereas our external obliques are more superior. They actually come up over the ribs and they actually interdigitate with the serratus anterior. Now, I'll show you the superior and inferior part of this in just a minute, but let's go ahead and draw them in. I'm not going to draw them overlapping the same way we were talking about before because I want to be able to see them all. But over here, our external obliques, like we said, are going to come down and in. And our internal obliques are going to go down and out. So notice we basically have 4 groups of muscles at 4 different orientations, all working together to form this protective barrier. All right. Questions on the anatomy of this?
I have a question. So is our abdomen one of the fewer areas that has this many muscles going on, or is it a pretty common thing, this layering of 4 different kinds?
Well, again, it is more unique in this area because you have 4 broad layers, right? In your forearm, you have dozens of muscles. You have well over a dozen muscles in your forearm to help you manipulate your hand and your fingers. So you have a lot of muscles in a very small compartment in this space, right? But they're very small bellied muscles that are all kind of nestled together in this. In this case, you have big, large sheets of muscles that are here. So absolutely, this is somewhat unique in that these big, broad sheets of muscle. So it's not uncommon to have lots of muscles in the area, but having big, broad sheets of muscle like this are somewhat different. All right, I want you to notice one other thing. When you look at the actions of the internal and external obliques, what do you notice about their actions?
They do a lot of the same, they do all of the same things.
All of the same things. Their actions are completely identical. Notice, being at angles, they cross the vertebrae at all these angles, allowing them to twist and turn the vertebral column a lot. But let's look more closely. Notice in this case on this side, and I'll go ahead and circle it in black. Notice over here, for instance, we have one of the external obliques with the muscle angled from the top left down to the top right, because we know it goes down and in. But notice over here on our right side, the muscle that goes from the left to the right would actually be the internal oblique. Notice over here, the internal obliques, because they go down and out. The external oblique on one side is angled the same, direction as the internal oblique on the other. And the internal oblique on one side is angled the same as the external oblique on the other. So it's not surprising that their actions are identical, because if you think about it, hold on. if, for instance, I wanted to twist my vertebral column to the side, or more appropriate, rotate the vertebral column, notice I would use the external oblique on one side coordinated with the internal oblique on the other. So because of the way they're oriented, when you're twisting, when you're turning, you're basically using the internal on one side and the external on the other, or vice versa. So not surprisingly, their actions are identical. And what are their actions? How many actions do they have?
A lot.
Reactions?
4.
4. Excellent. What are they?
Compress the abdomen.
All right. Suck in the gut. What else?
Second.
Flex.
And laterally.
Laterally flex.
And
Laterally
rotate.
flex.
vertebral
So
column.
flex, laterally flex, compress the abdomen, and what's the other one?
And rotate the vertebral column.
Rotate. Excellent. Anybody come up with a good mnemonic to help us to remember all 4 of those together? When would you do all 4 of those together?
Or am I wrong?
I'm sorry?
Like a, I know I could be wrong. A hula hoop?
Okay, I definitely could see most of the actions. I'm not sure that you're necessarily, oh, you would flex, laterally flex. Do you rotate your vertebral column when you're doing that?
Oh no.
But I think all the
What
other ones,
about- Like a bicycle crunch?
yeah, that's the one that I think most people think of, right? Because again, not only are you doing the crunch, but you're twisting to go across, right? To touch your left elbow to your right knee and vice versa the other way. So I think that definitely is, probably the classic example of, and not surprisingly, one of those exercises you do to strengthen these muscles, right? Because that way you're engaging all of the muscles, right? The rectus abdominis, the transverse abdominis, and both sets of obliques. Again, my drawing for this is truly amazing, but if we look at the pictures from your textbook, I think there's some really great things to see here. And then we can finally get to Yulia's question about the hernia. So here we see the abdominal muscles, the abdominal girdle. Notice first the rectus abdominis with those tendinous insertions, giving us again 1, 2, 3, 4, 5, 6, 7, 8 bellies for that 8-pack, not just the 6-pack, but an 8-pack. Absolutely from that. It's the one that is most medial. Notice our deepest one over here is the transverse abdominis, where we can see the fibers going horizontally. Notice here, I think we use blue for it. We have the internal, where we can see the fascicles going down and out. And notice they only come up to the edge of the ribs, whereas here is the cut, and here we see it intact. We see the external oblique, where the fascicles are coming down and in. Notice it goes over the ribs, and notice as we see, it actually interdigitates, it interlocks with the serratus anterior. Again, this does a nice job of showing it, but once again, your book does this great job of, hold on, there we go, separating the individual muscles as well. Notice here, we can again, very, very clearly see the orientation. Rectus abdominis goes vertically. Transverse abdominis goes horizontally. Internal oblique goes down and out, and notice just barely gets to the ribs. External oblique goes down and in, and goes way up over the top of the ribs as well. So here we see them individually. And the other view that we have from your textbook that does a really great job of showing this is a cross-section view. Notice with the cross-section view, there's actually 3 important things that we can see here. We see that the orientation of the bellies. Again, here we see the 2 rectus abdominis muscles. Here we see the 3 layers of the lateral part, External oblique is the most superficial. Let's do that. External oblique is the most superficial. Then the internal oblique, and the transversus abdominis is the deepest. But I want you to notice something else. These are big, broad, flat muscles. And so not surprisingly, big, broad, flat muscles have big, broad, flat tendons. And didn't we have a fancy term for a big, broad, flat tendon? Well, the answer is yes. So I'll ask the question this way. What is the fancy word for a big, broad, flat tendon?
Aponeurosis?
Aponeurosis, absolutely. And notice each of these muscles has an aponeurosis. The superficial, the medium, the deepest one. Notice it's just the external that is on top. So if we go back to that original picture, notice over here on the right-hand side, that aponeurosis of the external oblique is intact. We can see this big sheet of connective tissue that is sitting on top of the muscles. And as we look through the cross section, we can see how this works. Notice how all of the individual aponeuroses all merge together, wrap around, and support the rectus abdominis. And it is the aponeurosis of the external oblique that is on top. And here's the last thing I wanted to show you from this illustration. Notice that all of the aponeuroses from the right side and all of the aponeuroses from the left side all come together in the middle to form a thin line of connective tissue right at the midline of our body. And so again, if we go back to that original picture, notice right here on the midline of the body between the two rectus abdominis is Jessica Alba's younger sister. And what do we call that thin white line at the midline of the body? Linea Alba, right? Linea Alba literally means linea, line, alba, white. It's the white line, right? This white line is where the, This white line is where, and let's use that and use that. This white line right down the middle is where all these connective tissues come together and merge. Now, it's still not muscle holding them together. Yes, Laura, did you have a question?
Yeah, is the linea alba based on its location? Because we see like higher right by the pectoralis major. There's also one.
Correct. You are correct. The linea alba is just between the abdominal muscles. So it's not this connective tissue, right? It's not the connective tissue between the pectoralis majors. It's just here associated with the abdominal girdle. Right? Now, this white line, again, is where the connective tissues come together. However, again, these are collagen fibers. And as we know, they don't stretch very well. So one of the things that happens, especially with a fast, rapid enlargement of the abdominal region, like when a female is pregnant, as the belly expands, one of the things that happens is that these fibers can be stretched out. And so what can actually happen is, especially below the umbilicus, below the belly button, the thinning of those tissues can make it a little bit more transparent, so you can see some of the darkness. It's not like you're going to see the kid waving from inside, but what ends up happening is a dark line forms typically below the belly button on a pregnant woman, especially late term, from the stressing and the stretching of those threads, what is commonly referred to as the linea nigra, the black line. That typically goes away shortly after pregnancy as everything starts to bounce back. But to get back to the original question, right, the abdominal pelvic cavity is a place where a tremendous amount of pressure can be produced. right? When you're trying to lift up weights, when you're trying to defecate, when you're trying to, do all sorts of other things, you can produce a tremendous amount of pressure. And if you produce too much pressure, that pressure will try to find a weak spot to work its way out. And that weak spot is commonly the umbilicus. And what can happen is those connective tissues can stretch be stretched out by the fluid inside, or maybe even some of the tissue from inside the abdominal cavity can bulge out in that region. And even though someone mentioned it earlier, what do we typically call that condition?
A hernia.
Yeah, an umbilical hernia. Absolutely. And last time I checked, both boys and girls have belly buttons. So both men and women are equally likely to get umbilical hernias. All right. Questions on that? I make a point of, oh, go ahead. Yes.
I'm just curious, sometimes after people have a baby, they have, I think it's called like diastasis, something where their abdominal muscles separate and they have to have like a surgery to put them back together. Would that be a muscle, or it would be the tendon that occurs?
I'm afraid I'm not familiar with that condition. It's not my area of expertise. My guess, based on the name of it, is probably the connective tissues, the aponeuroses, have been stretched in a way where the muscles have been displaced in relation to each other. And so typically it's those, because again, they don't stretch well and they don't have elasticity. So it's hard for them to go back into place. So my guess is that it's a displacement of the muscles caused by a stretching of the aponeuroses of their tendons.
Yeah, I had it, and my doctor just said don't do any strenuous ab exercises until you can feel that. You could actually put your finger in it and you feel muscle separation until you can feel it go away in less than a centimeter.
Right, because again, doing those abdominal exercises dramatically increases the tension, and that could again cause more separation to occur. So absolutely. Wowzers. All right. Notice there's one more structure here. So we've hit two structures so far, the aponeurosis of the external oblique, the linea alba, but there is one more important structure here as well, and it is the inguinal ligament. The inguinal ligament, and actually I think where we see it best, let's go to these individual pictures. Although this one shows it really, really nicely. Here, let's start here. Inguinal ligament goes from the superior anterior iliac spine to the pubic tubercle. So we can see that right there, but I think where we see this best, perfect. Notice, look at this picture. On all of these pictures, and again, you don't need to know the origins and the insertions for these muscles, but notice all of these muscles and their tendons are all associated with this ligament that starts at the superior anterior iliac spine and goes to the pubic tubercle. This ligament right here is what is known as the inguinal ligament. It is an attachment point for all of the abdominal muscles, so it helps to support your abdominal girdle. However, and then this is the other thing that I want you to see about it, is that in both males and females, there is a space there known as the inguinal canal. In males, the spermatocord, which carries blood vessels, nerves, and sperm from the testes back into the abdominal pelvic cavity, pass through that space. In females, this inguinal canal is just a potential space. What do I mean when I say potential space?
It's only used sometimes, like when
Sort of. Well, think of it this way.
It may or may not be there.
How many hands do I have here? Not your question. Two
Just
hands. If I smash them together, is there a space in between them right now?
small, tiny.
Okay, tiny, but really, yes, because
It's
it's
a
not. But
chemistry.
if I was able to perfectly smash them together, there would be no space there. Would that mean they just became one hand? They just became one thing? No, it would still be two things smashed together. There would be the potential for a space between it, but for all practical purposes, there's no space there and nothing in it. And that's basically what it is for women. In women, there's just a potential space there, but in males, that spermatocord has to pass through. I make a point of emphasizing this because we've been talking about how you can build up all this pressure in the abdominal pelvic cavity. And one place it can try to escape is the belly button, forming an inguinal hernia. But in males, where this spermatocord goes through the inguinal canal, this area is also a weak spot where pressure can force and bulge its way out. And what do we call that condition?
Cancer?
Not cancer, an
hernia?
inguinal hernia. It's an inguinal hernia, right? Males can have an umbilical hernia or can have an inguinal hernia. Most inguinal hernias, over 90% of inguinal hernias occur just in males. It is much less common for it to occur in females because it's just a potential space. Very rarely will it happen in females. It's not impossible, but very, very rarely. Most inguinal hernias occur only in males. Whereas, like we said, umbilical hernias are equally likely in males and females because both males and females have belly buttons. Only males have spermatocords, so only males have an open inguinal canal in their inguinal ligament. And so their males are much more likely to get an inguinal hernia than females are.
I have a quick question, but it's
Yes.
going to slightly backtrack. I didn't write down the definition of the aponeurosis of external oblique. How did you define it?
The aponeurosis, remember, aponeurosis is a big, broad, flat tendon. This big, broad, flat tendon on the front of the abdominal region is the aponeurosis of the external oblique.
Thank you so much. I got it down now.
Excellent. Exactly. The things we do just to produce gametes, it's exhausting, horrible work. We shouldn't really, males shouldn't be required to do anything else. All right, excellent. Questions on that? All right, there's one more structure I want to identify for you as well. And that, again, it's all in the name. Thoracicolumbar, thoracic and lumbar region of the body. Fascia, it's a connective tissue. So notice there is this white triangular region on the posterior part of the body. It is not a single tendon. It is a fascia. So it is a bunch of dense irregular connective tissues supporting multiple muscles and holding them in place. So that's why on the front it's an aponeurosis. It's a single tendon of a single muscle. Here it's a fascia because it is supporting many muscles together.
Did you say irregular or regular connective tissue?
Dense irregular connective tissue. It's a fascia. It's a dense irregular connective tissue. All right, and that's, I think, the last structure on your list that you are responsible for there. Right? Questions on that? Was that it? Don't make me look at my list. Someone tell me, were those all of the structures? Okay, excellent.
So there's 4.
Yeah, Linea Alba, aponeurosis of the external oblique, inguinal ligament, and thoracicolumbar fascia. Those are the 4.
I have a quick question. Did we, for some reason, I have levator ani as on mark. Did we cover it last time?
Yeah, remember the levator ani is a muscle group that was part of the urogenital diaphragm.
Oh, okay. Okay, I made a side note of urogenital, but I didn't realize it's that muscle.
Yeah, it is the primary muscle of the urogenital diaphragm. Yes. And remember, it's a muscle group, but you don't need to know the names of the individual muscles that form it. All right, excellent. Where are we on time? Just that simply, we are perfect. We are through all of the muscles of the trunk. So now we can move on to the muscles that move the arm through space. And let's start again first with the ones whose origins and insertions we are responsible for. And let's talk first about typically what is the most misnamed and most common muscle, right? Everybody's aware of the biceps, right? You do the curls for the girls, so you can get those nice big biceps, as we were talking about. However, it's not the biceps. Biceps just means two-headed. You have over 4 biceps in your body. You're responsible for at least 2 of them on this exam, so we need to be precise. It is the biceps brachia. It is the two-headed muscle in the brachial region of the arm. And as we look here, we can clearly see that there are indeed 1 and 2 different bellies. So this has 2 bellies or 2 heads. And if it has 2 heads or 2 bellies, and again, either term is acceptable, what else does it have?
2 origins.
2 origins. Excellent. Absolutely. So on the exam, if I have an arrow like this and say identify the muscle, and I have an arrow like this, actually let's switch that, and ask you to identify the muscle, are the answers to that the same or different?
The same.
Different. And how am I going to emphasize to you that these are different questions on the exam? I'm going to say, what is it?
Whichever is more medial.
Well, no, that's how you tell them apart. But what am I going to say on the exam? Identify the muscle. What's that magic phrase that I told you to keep an eye out for that make the warning bells go off so that you can tell them apart? That I'm wanting
Left and
something
right.
special. Nope, although that's a good guess.
Specific.
There you
Medial?
go, be specific. So if I say identify the muscle indicated by the green arrow, be specific, or I say identify the muscle identified by the blue arrow, be specific, these are not the same questions and these are not the same answers. Now, anybody happen to know what the be specific muscle that is the blue arrow is?
The short head.
Well, you got a 50-50 shot, so is it long or is it short?
Long.
It is indeed the long. The way I actually remember it myself is the long is lateral. So L long, L lateral, that's how I tell them apart. So the long head is lateral and it is the short head.
It doesn't look much longer or shorter from the picture.
Exactly, because when they typically talk about a muscle being long or short, they're not actually talking about the muscle. What they're actually talking about is their tendons. Notice if you look closely, you're absolutely correct. The bellies of these muscles don't look that different. But if you notice, the tendon for the short head basically comes straight off the arm right up to the coracoid process of the scapula. Whereas the tendon from the long head travels through the intertubercular sulcus, goes over the head of the humerus, into the joint cavity of the humerus, all the way to the supraglenoid tubercle of the scapula. So it has the much longer tendon. So the long head, oh, but I totally colored those wrong, didn't I? Whoops. The blue is, No, I have this right. Blue is the long head. There you go. So I did do that right. Okay. Oh, this is what's colored wrong. That's what screwed me up. And then this one is the short one because it just goes to the coracoid process. So yeah, it's actually the tendons. When they say long and short, in this case, it's really the tendons that they're looking at. All right.
So would the answer would be the long head of the biceps brachia? That
Correct.
would be
So
how you.
if and again on an essay question you've seen, I'm not going to have two arrows. I have two arrows here so we can do it at the same time. But I could easily have this picture of this arm with the green arrow, and I could ask you the question, identify the muscle, be specific. And what I'm asking for there is the short head of the biceps brachia. Or if I say identify the muscle indicated by the blue arrow, be specific, then I'm looking for the long head of the bicep brachia. Right?
Okay.
If you just
I think I was getting confused.
Yeah.
Oh, sorry. I think I was getting confused with bones with head, like more of like a location on the muscle versus the entire muscle name.
Yep. No, it's the entire muscle name. Absolutely. And again, this is the phrase that is the dead giveaway. I'm looking for something special when I say be specific. Because when I say be specific, I mean be specific. If you just said bicep brachia, you are not getting it correct. If I say identify the muscle indicated by the green arrow, and you say the short head and the long head of the bicep brachia, you're not getting that correct because you were not specific. I want just the head that I'm pointing at, and that be specific is my way of telling you that. And remember, not only does it work for the names, but it also works for the origins. So let's do that. Let's draw the origins of the bicep brachia. Excellent. So let's start easy. What is the origin of the short head? Hold on. Red 14.
The coracoid process.
Excellent. The origin of the short head is indeed the coracoid process. So we'll use that red. We'll use a small line. And remember, that is that bird's beak that sticks off of the scapula. You know, one of the questions that I love, notice this is an important attachment point. One of the kind of questions I love to have on the exam, and again, this doesn't work on yours because it's normally something in a station-based exam like we would normally have. You'd come to a station and that scapula would be sitting here, and I would have a scapula with a tag on the coracoid process. And question 1 would be identify the muscle that originates from this bone feature, be specific. And what would your answer to that be?
Short head.
Can you get partial credit for that?
Short head of bicep brachia.
Excellent. There you go. No, short head.
Right arm.
No, just short head of bicep brachia is what I'm looking for. Absolutely. But notice I could have that scapula there with that coracoid process tagged. And then the very next question could be identify the muscle that inserts into this bone feature. And what inserted into the coracoid process of the scapula?
Can you say that question again, please?
So on the exam, and I guarantee you this is a question that's in the pool of lab questions. I guarantee there's a picture of a scapula with a tag pointing to, or an arrow pointing to the coracoid process. And the question is, identify the muscle that inserts into this bone feature.
Wouldn't it be the long head because it kind of goes past it?
Nope. What attaches to it and inserts there? Oh, there you go. Amanda's got it. Pectoralis minor. Notice this is the origin for the short head of the bicep brachia, and it is also the insertion for the pectoralis minor. Some of these bone features are used for multiple things. Excellent. All right. And what did we say the origin? Let's do this. I'll use pink just to separate it. What is the origin of the long head again?
Supraglenoid tubercle.
Excellent. So again, right up here above the glenoid cavity on the very top of our scapula, right up there on the scapula is our origin of our long head. And so that long head, its tendon again comes across the head, down the intertubercular sulcus, and then we have the long head here. Our short head comes straight down, and we have the short head here. So side by side, long head, short head, side by side, origins of the long head, origins of the short head. And again, on the exam, if I have an arrow pointing here and I say, identify the origin of this muscle, be specific. And in that answer blank, you write, the origin of the long head is the supraglenoid tubercle of the scapula, and the origin of the short head is the coracoid process of the scapula. That information is 100% correct. So are you getting that question correct?
Yes.
No.
No.
Because I told you to be specific. Be specific means tell me just for the muscle belly I'm pointing at. I don't want all of the origins. I want the one specifically for the head I am pointing towards. All right. Again, one of the things I like to do is give partial credit to help you guys to be successful in this learning process. But if you see those magical words, be specific, then that is exactly what I'm asking for you, and I'm going to be a hard ass about it. I want a very precise piece of information, so make sure you just give me that very precise piece of information. And again, be specific is my way of warning you that I want a very precise piece of information. So there's no ambiguity. You will know when I want it because it will be very precisely worded that way. All right, now we have the origins, the two origins, we have the two bellies, but how many insertions do we have?
Just one.
Just one, excellent. And what is it?
Common tendon into the radial tuberosity.
Excellent. The radial tuberosity, excellent. And again, one of the things that I want to emphasize is as you read the handout, I have said a common tendon into, just to remind you that the two bellies come together. If I ask you for that on the exam, you don't have to say, I mean, look, there's nothing wrong with saying by a common tendon into the radial tuberosity, but if I ask for the insertion, all you have to say is the radial tuberosity of the radius. And heck, you don't even have to say of the radius because it's in the term, radial tuberosity. It's the tuberosity of the radius. So that's all I'm looking for. All right, so there you go. Those are our two origins. That is our one insertion.
Did the origin of the short head, is it same as the red? That's why I didn't get a second mark.
No, they're not the same. They're just next to each other. This one is red here. That's the short head. The pink is over here. What if I use a different color? What if I use green?
Oh, okay. I see it now.
Oh yeah, this little thing right here, the top of the scapula, the top of the glenoid cavity above the scapula is that opening there, is that bone feature there. Okay. All right, and that's the long head. Excellent. So notice one interesting thing about the bicep brachia. The bicep brachia is a big superficial muscle where the two bellies are sitting side by side, right in the brachial region, sitting on top of the humerus. But does it actually attach to the humerus?
No.
Yeah, it doesn't actually attach to the humerus. So how many joints does this muscle cross?
One, I think.
Two.
Two. What two joints?
Your shoulder and elbow.
There you go, shoulder and elbow. So it affects both the shoulder and the elbow. Now, bicep brachia is an anterior muscle, And in general, as we know the joints work, what do almost all of the anterior muscles do?
Have a
Flex.
posterior, oh, flex.
So what's the effect that it has on the shoulder? Flexes the shoulder. What effect does it have on the elbow?
It flexes the elbow.
Flexes the elbow. So there you go. Then remember, we have something interesting going on in our forearm. In our forearm, we have this ability to rotate the radius so that my hand can point forward or my hand can point backward. What kind of actions did we call that again?
Supination, pronation.
Pronation and supination, absolutely. Pronation and supination. And remember, it's the radius that moves that pronates and supinates. So notice if a muscle attaches to the radius, it can affect pronation and supination. If it affects to the ulna, since the ulna doesn't move, it can't. And with our bicep brachia, does it affect to the movable bone or the stable bone?
Stable.
The ulna is stable. Is it attached to the ulna?
No. The movable.
Movable bone.
So what is our third action of the bicep brachia?
Supination.
Supination. It supinates the forearm. So flexes the elbow, pardon me, flexes the shoulder, flexes the elbow, and supinates the forearm. 3 actions of the bicep brachia. Did anybody come up with anything where you might use this muscle like that?
Bowling.
There you go. Absolutely. Classic example, bowling, right? If you think about when you're throwing that bowling ball, you're using your shoulder, you're putting the spin on it by using your elbow, and you have to have your hand supinated so that you can hold the ball, right? Classic example. That bowling is a perfect example of something you would do with that bicep brachia. So there you go. Origin, insertion, actions. Questions on the bicep brachia?
You said bicep brachia is one of the most commonly mislabeled.
Misnamed. You grab some random person on the street and you ask them to identify a muscle in their body. They would typically point to their upper arm and say, my biceps, right? Because I work out at the gym to do the curls for the girls so I can woo women with my big, huge, massive biceps, right? And if you just put biceps on the exam, are you going to get that right?
No, we have to say biceps brachia and then which
yeah,
one?
Exactly. Because we've also got a biceps femoris. And like I said, you have several other biceps as well. Ones you're not responsible for on the exam, but bicep brachia. So I was just saying that it's a muscle that most people know, but most people misname. That was what I was trying to point out. That was the point I was trying to emphasize. Okay. So I want you to notice one other thing about the bicep brachia. As I mentioned, we keep talking about the curls for the girls. So you get this nice big muscle because it is this big, huge, right, parallel muscle. So those are the ones that get fatter when they contract, where the two bellies are side by side here, and they're superficial on the anterior side. But if you look at this illustration really closely, what you will notice is deep to the bicep brachia on both sides is a muscle that really helps the bicep brachia to do its job. And that muscle is the brachialis. It is deep to the bicep brachia and hard to see just from this anterior view, but it's sitting nicely behind it. However, while it sits right behind the bicep brachia, it has a very different origin and insertion. So not surprisingly, its actions are going to be different as well. So let's take a look at that. What is the origin of the brachialis?
Fred distal half of humerus.
Yeah, basically, let's actually make this a little bigger because it'll be easier for me to draw it. Right. Basically, it is the diaphysis The anterior diaphysis of the humerus. And what is its insertion? Actually, hold on, I need to make that a little higher. There you go, about right there. And what is its insertion?
Coronoid process of ulna.
Yeah, notice right here on that coronoid process of the ulna. Now, notice it sits right under the bicep brachia, but how many joints does the brachialis cross?
Just one.
Yeah, just one, the elbow. And notice, does it insert on the movable bone or the stable bone?
Inserts on the stable, I think.
Yeah, ulna is the stable bone. So it can't pronate, it can't supinate, it only crosses the elbow, and it's an anterior muscle. So what is the one and only one action it has?
Flex.
Right. Absolutely, just flexes the elbow. Right. So it just flexes the elbow. So even though it sits right underneath the bicep brachia and shares that ability to flex the elbow, it can't pronate or supinate. It doesn't affect the shoulder because of its origin and its insertion. Now, there is one interesting thing about this. We haven't gotten to the deltoid muscle yet. But as we know, the deltoid muscle is this big, huge lateral muscle that sits on the lateral aspect of the arm, like a big, huge, fat triangle. So we have this big, huge, fat triangle that comes over the top. And what's interesting about the deltoid is when the deltoid comes down, and here's another fancy technical word we're going to use, it embraces the brachialis. Embracing basically means that the two muscles come together. Where one inserts is where the other one begins.
Oh, yeah. Catch bird.
Yep. All right. So embraces means where one inserts is where the other originates. They come and connect to each other. This is important because even though the brachialis is a deep muscle, oops, wrong buttons. That's all right. We muted it. Even though it is a deep muscle, notice that no matter how big that arm gets, that embracing stays there. So notice, even though the deltoid muscle is big and huge and massive, notice the point of it still comes and always points at the brachialis. So notice here, we have the long head of the bicep brachia. And right here, deep to it, we have the brachialis. Same thing over here. The head of the deltoid always points right at the brachialis. So even though it's a deep muscle, it's one we can very easily see on our bodybuilders. So it will be one of the muscles. So I said you're responsible for superficial muscles, but this one, because of its so obvious location, it is one that you will also be responsible for on your bodybuilders as well. And since we're doing it, let's go ahead and do it here.
I have a quick question.
Yep.
So on this model, the two bellies of the brachii, they're separated, even though normally they lie pretty close together, and we don't see.
Well, notice he's got his arm turned. So the way he's got his arm turned, we're just seeing the lateral belly. So here is the lateral belly of the bicep brachia because his arm is turned to the inward. Notice his arm is turned inward.
Oh, okay, yeah, he's not in an anatomical position.
Right, exactly. Notice also, so that is the long head of the bicep brachia. This is the brachialis. And even though we haven't done it yet, does anybody happen to know what this big, large belly over here might be then?
Brachial radialis.
Tricep.
Try.
Yeah, exactly. Notice because he's got his arm twisted, you're looking actually at the back of his arm, and that is actually the lateral head of his tricep brachia. So because he has his arm twisted, we're seeing the front, the side, and the back all at once. And that's how we're able to see all three of those muscles. And see, no matter how big these arms get, you can still see these three muscles separated from each other. And the deltoid always is going to point to the brachialis. So it makes it really nice and easy to identify, right? Notice we go back to our, mere mortal body. They've left the head of the deltoid. They've removed it so we could see all the tendons, but we could still see how that head of the deltoid points right at the brachialis. All right. I think it's more just the angle that he's got it turned at. You're only seeing one of the three heads of his tricep there. And I would argue that I would think most people's biceps are bigger than their triceps because most people like doing those exercises more than the tricep. But again, you're only seeing the one belly. You're not seeing all of it. All right. There is one more important flexor of the elbow. And when you first look at it, it isn't a muscle you would expect to be a flexor of the elbow because it's down in the forearm. Forearm muscles, we typically think of being muscles that affect the wrist and affect the hand. And so here we have this muscle, the brachioradialis, that looks like it might be a forearm muscle, but notice when we look at the origin and the insertion, we can see what it really does. So give me that. What is the origin of the brachioradialis?
Lateral supracondylar ridge of humerus.
Excellent. So that lateral supracondylar ridge, that raised ridge on the lateral aspect of the humerus. And what is the insertion?
Styloid process of radius.
There we go. Styloid process of the radius. Notice, even though this is a forearm muscle, does it go across the wrist? Does it affect the carpals? Does it affect any of the bones of the hand?
No.
No. In fact, how many joints does it cross?
One.
Just one, just the elbow. Of course, does it go to the movable bone or the stable bone?
Movable?
Movable. So does it pronate or supinate?
Both.
Both. How can it do both? No muscles both flex and extend the elbow or flex and extend the arm. How can you both pronate and supinate? Anybody know?
Oh, is it because sometimes our muscles can switch their origins and insertions?
True, you are right. We did talk about how muscles can change their origins or insertions. But here's the key. Remember, one of the things we've been saying for a long time now is when you're in anatomical position, most of your muscles are at their normal, natural position. And of course, the key word there is most, because most isn't all. After all, if you're standing waiting for a bus, do you stand there in anatomical position like this, waiting for the bus?
No.
No, if you did that, people would slowly be moving away from you in fear. Typically, when we're waiting for the bus, we hold our hands in what we call body neutral position. Body neutral position is actually where the thumb points anteriorly and the palms point medially. And that's actually what this muscle does. This muscle puts the hand into body neutral position. So notice if you were already pronated, when you contract this muscle, it would supinate it to bring to body neutral. If you are already supinated, when you contracted this muscle, it would pronate it to bring it into body neutral position. So notice what this muscle does is it crosses the elbow, so it flexes the elbow, and it can both pronate or supinate to bring the hand to body neutral. And that's what it's doing. It's bringing that hand to body neutral position. Right? So that's what this one does. You can actually see this muscle for yourself. What you can do, make a fist, put it underneath the edge of your table with your arm flexed. And as you push up, you will see that muscle that sticks out right on top of your arm. That muscle that sticks out right on top of your arm is your brachial radialis. In fact, if you think about it, it is a very common bodybuilder pose, right? That pose where they're basically pushing down on their arm to make their brachial radialis bulge out. Now notice our bodybuilder here has her hand turned, because notice the brachial radialis always lines up with the thumb. So she's got it turned, which is why it's not bulging straight out. Notice we go back to our previous bodybuilder, right? Again, if you look at where his thumb is and lines up, you can see that brachial radialis sticking out there. So again, you make that wrist, you push up on it, and you can see that muscle bulge out. That is your brachial radialis. And it lines up right with your thumb because its goal is to bring you into that body neutral position. All right. Questions on that? What activity? Did anybody come up with an activity for this one?
I have a quick question. I'm still unsure as to how it can both be an insertion and an origin and switch.
Okay, well, this muscle can't. We're not talking about this muscle when we do that. But
oh, okay.
Yes, this muscle can't. No. What
But
we're
it can pronate and supinate? It can do both?
The way it pronates and supinates is totally dependent on where the hand is located. If you are already supinated, so again, it wants to line up your thumb so that your thumb points anteriorly. That's what this muscle wants to do. So if you're already supinated, if you have your hand palm out, then it is going to pronate it to bring it to that 90 degrees. If you already have it pronated, it is going to supinate it to bring it to 90 degrees. So it doesn't care where it starts, it just cares where it finishes. So it has the ability to both supinate or pronate because it wants to bring it to body neutral position. All right. Yep. So again, it might have been trickier because you might not have fully understand how it could pronate and supinate. And again, a lot of times people think that this is the Beyonce muscle, right? Because all the single ladies, you got that arm flexed and you're pronating and supinating. And if it helps you to remember the three of them, then that definitely is worthwhile. The single lady, this is definitely the single lady muscle for that. However, I, again, always think of it in terms of bringing that arm to body neutral position and flexing it. Right. And again, as we've learned before, the whole reason to contract muscles is to woo women. So the way I wooed my wife, for instance, I saw her across the bar. And so to impress her, I whipped out my best dance moves. And of course, my best dance move is, of course, the robot. And how do you do the robot? You put your hand in body neutral position and you flex your arms. Right. And so there I was roboting and she just swooned as a result of it. So there you go. So oh yeah, getting ready to shake a hand, that's a good way as well. Absolutely, right? Because you're going to put it in the body neutral position and flex it to put it out there. There you go. That's a good one. Absolutely. Exactly. If you come over, I will tell you the full story of how I wooed my wife. All right, I won't be holding the breath. And I don't have cookies. I told you I don't bake. I cook. I don't bake. Baking is too much precision. I don't like precision. In the kitchen. All righty, excellent. So how are we on time? I think we can sneak in one or two more. Next, deltoid muscle. We kind of already talked about this a couple times. Well, yes, I talked about chemistry stupid. Deltoid is again, multipennate muscle that is a lateral muscle. But again, when we look at the origin of it, we can see how it truly is a lateral muscle with both its origin and its insertion. What is the origin of the, I guess I got to go back a page here. No, are you not going back? Oh, that's what I want. There we go. This is what I want. Forward, that's what the problem is. Okay, excellent. What is the origin of the deltoid muscle?
Embraces insertion of trap, acromial end of clavicle spine, and acromion end of scapula.
Excellent. It has the exact same origin as the insertion of the trapezius. Starts on the spine of the scapula, out to the acromion, and then onto the anterior part of the clavicle, the lateral third of the clavicle. So the same place where the trapezius comes in and inserts, it embraces that trapezius. And as our illustration shows nicely, it inserts into that lateral roughened up patch on the lateral aspect of the arm, that is that deltoid tuberosity. And so notice, and here I'll do them both in green. Our trapezius, as we talked about, inserts where the deltoid starts, so they embrace. And as we just learned, our brachialis embraces the deltoid where it inserts. Where it inserts is where the brachialis begins. So our deltoid is just nestled here in between the trapezius and the brachialis. So big lateral muscle crossing the shoulder, just one joint. We know anterior muscles flex. What's the obvious thing that lateral muscles do?
Extend,
Well, in this case, it's going to
Well,
rotate.
abduct.
yeah, abduct, right? A lateral muscle is going to bring the arm away from the body. So the most obvious thing it's going to do is an abduction, right? That is the most obvious thing that it is going to do. But you do have the right idea. Notice some of its fascicles are located on the anterior part. So if we just flex the anterior part, basically we could turn the humerus so that my arm rotated inward, right? So if you think about what's going to happen to the humerus, I can do a medial rotation by just contracting the anterior part. And notice if I just contracted the back part of my deltoid, I could laterally rotate my humerus. So I can medially and laterally rotate my humerus by using just some of the muscles, or if I use all of them, I can abduct it. So again, 3 actions to this muscle: abduction and a lateral rotation and a medial rotation. Anybody come up with something clever for this one?
I always think of the Delta Airlines and holding onto the railing as you try to find your seat on the airplane.
Well, you definitely have to be abducted to that so that you can work your way through the aisles. I'm not sure how much medial and lateral rotation you would do for that.
You put the luggage away.
Oh, there you go. Okay, I like that. Absolutely. Or something like that. You want to slap people on the heads as you go by. Excellent. I think that's a pretty good one. For me, as I mentioned, I did both my undergraduate and graduate work at Davis. So I spent far too much time in Davis, more time in Davis than any sane person should have to today. And one of the things that happens if you live in Davis, you are actually required by law to spend at least 90 minutes a day on your bicycle. And so when you're out riding your bicycle around, one of the things you have to be able to let people know is where you're going, right? Of course, how do you do that? You put your arm out, and if I'm going to turn right, I would laterally rotate my arm so that I could go that way. And if I'm going to stop, I immediately rotate. So those bike signs that you give, right, when you're on your bike are examples of using this muscle. Abduct, and you immediately and laterally rotate. Yeah, there you go, exactly. Required by law. Oh, it's not an exercise. They just have to make you do it to get around. Yep. See, there you go. The people are there. Right. Again, tunnels for the frogs And bicycles for the people. That's right. Instead of a chicken in every pot and a car in every garage, they a tunnel for every frog and a bike for every person. Davis is a magical place. All right, excellent. Questions on that one? I can tell when it's getting late in the lecture because everybody gets way more punchy in the way, including myself, in the way things that we're talking about. All right. Yeah. All right. We're behind, so we got to get these two in. All right. 2 more, and then we'll call it a day. The one that was drawn before, it should say right there is the trapezius. And that's why I put the trapezius and the deltoid on the same one, is so that you can see how their origins and insertions embrace. Right? That was the whole point of having that. And again, you can always print out multiple pages if you want, or do it again if you need to, or anything along those lines. All right, there are 2 more muscles I want to get through before we finish for the day. The first is the latissimus dorsi. The latissimus dorsi is a superficial, inferior, so let's actually write this down. Mostly posterior muscle. Kind of similar to the trapezius. Trapezius was a superficial, superior, mostly posterior muscle, where this is a superficial, inferior, mostly posterior muscle. And again, the easiest way to see this is with its big, huge, fancy origin. What is the origin of the latissimus dorsi?
Thoracolumnar fascia.
Excellent. So that big connective tissue we know back there. But what are the bone ones?
Spines of lower 6 thoracic vertebrae, lumbar vertebrae, lower 3 to 4 ribs, crest.
Okay, hold on. Slow down. You got it all. Excellent. So T12, 11, 10, 9, 8, and 7. So T7 through all the lumbar. So through L5. Bing, bang, boom, bomb. So excellent. We have that. Where else?
That was the lower 6 thoracic, right?
Yep. Lower 6 thoracic, the spinous processes of the lumbars. What else?
Lower 3 to 4 ribs.
Out here a little bit on the ribs. What else?
A crest of sacrum and ilium. So
the crest of the sacrum back here, and the crest of our ilium up here. And one more, right?
And yeah, inferior angle of scapula.
There we go. Notice there are fascicles from this that start all over the place. This is a big, huge convergent muscle, right? Where we've got some fibers that are starting from way down here, some that are starting from up here, some that are starting closer, some that are coming across the ribs. But all of these come together into one single insertion. And what is the one single insertion for this muscle? Again, these answers should be right in front of you guys. These should be the easy ones to answer. All you have to do is read it off a list.
Intertubercle sulcus of humerus.
Yeah. Remember, and notice, I'm going to cheat and take advantage of it over here on the pectoralis minor picture. Remember, we have the greater tubercle, the lesser tubercle, and there's that indentation in between the 2 tubercles on the anterior side of the bone. And so notice I'm going to cheat and do this as a dotted line here. To remind us that that's on the anterior side. So it's on the anterior side of that. So it actually comes up under and attaches up here. Again, I'll show you in a second, but let's go ahead and do this. So again, whether it's from here or coming across from here coming from here. All of these all come together into that single insertion on the anterior side of the arm. So here we've drawn them, but let's look at some of the pictures that do a nice job of showing it. Notice here, we can see again this big, huge, broad muscle starting from T7 all the way to the sacrum, coming off of the spine, coming off of the inferior angle. We can't see where it's attaching to the ribs, but they're here. All of these are coming together. And actually where I think we see this best, where was that? Let me find that picture. That'll be nice. Oh, damn, I'm clever. All right, it was actually the very next slide. I had already done that, so that's always nice. Excellent. Notice here with our chart. Notice here when we look at our chart, what we can see is where those fibers of the latissimus dorsi are coming off of the back of the body to attach to the arm. Notice with his arm up, we can see where it's coming in and attaching to the anterior side. So it's actually wrapping around and coming up and attaching. This is the latissimus dorsi. Now, let's go back to the previous picture because I want us to see how this works. Notice, okay, so questions on its origin or its insertion. All right. Our goal, remember, for a muscle is to bring the insertion to the origin. Now, this is a posterior muscle. Being a posterior muscle, what do most posterior muscles do?
Hyperextend.
Well, or just extend. It doesn't even have to hyperextend, but you've got the right idea. They're going to anterior muscles flex, posterior muscles extend. So this is going to extend the arm. Notice it's also a muscle that starts on the midline. So what that means is it's going to bring the insertion towards the midline. And what kind of action would that be when we bring the insertion towards the midline?
AD.
Adduction. Excellent. Oops. Absolutely, adduction. Excellent. So I think those two are super obvious. But notice the third thing. Because it comes up to the arm on the front, when it pulls on the arm, it is going to turn the humerus so that the anterior part of the humerus goes towards the midline. So because of its anterior or insertion, it is going to medially rotate the arm. So by looking at this muscle, we can see what it does. It is going to adduct the arm, it is going to extend the arm, bring it back, and it's going to medially rotate it. This should have been a pretty easy one for you guys to figure out. What is a good action that involves the latissimus dorsi? Yeah, question.
Yeah, I have a quick question.
Yep.
So if on the exam we need to identify insertion and origin, would any of the correct origins be acceptable, or do we have to list more than one?
All.
We have to list all.
If I ask for the origin, I want the entire origin.
Okay.
Yep. Now the good news is if you give me at least one of those, you will get partial credit. But for full credit, I want the entire origin. All right. So did anybody come up with something clever for what you would use your latissimus dorsi for?
I was thinking pull up, but to medially rotate, maybe a chin up. Remember not really be.
I kind of like that. I could kind of see that. It's not, if you were doing it out and kind of bringing it in, that would be probably pretty good that way. Think of it this way. Who has really, really huge latissimus dorsi muscles?
Bodybuilders.
True, bodybuilders. Bodybuilders, all their muscles are big. Who else? Swimmers, right? Excellent, right? Michael Phelps practically has bat wings, right? Because think about it. When they're doing many of their strokes, right, they're bringing the arm in, they're bringing the arm back, and they're medially rotating, right? If they're doing them both at the same time, they're doing the butterfly. If they're alternating, they're doing freestyle. But that motion of bringing the arm in, bringing the arm back as you're medially rotating it, exactly, that's a big, huge swimming motion. Right. So again, Michael Phelps has massive, massive latissimus dorsi. And I'm told his sternocleidomastoid is very well developed as well. All right. Questions on that? All right. The reason I wanted to stay and do this one is because even though it doesn't seem like it, this latissimus dorsi relates very, very closely to the pectoralis major. Now, the pectoralis major is another convergent muscle, one of the ways they are similar. However, it is, again, superficial, same that way. It, in this case, is superior, and it is completely anterior. But when we look at its origins and its insertions, we're going to see some similarities. So let's do that. Let's look at the origin and the insertion of the pectoralis major. What is the origin of the pectoralis major?
Clavicle, sternum, cartilage of ribs 1 through 6, and aponeurosis of external oblique muscle.
Excellent. So notice superiorly, it attaches to the clavicle. Medially, it attaches to the sternum and the cartilage of the ribs. And even inferiorly, it actually attaches to that aponeurosis of the external oblique we've talked about. Notice it has this big, huge C-shape origin. And again, this is one of those muscles where you haven't thought of it in these terms, but if you think about it now, it makes sense. As these bodybuilders work out this muscle, and the muscle gets bigger and bigger, because of its origin, can it go up? Can it go down? Can it go medially? No, pretty much the only way it can grow is out. So when our bodybuilders work out, they get these big, huge, massive pectoralis major muscles that stick out because out is the only direction they can grow. They're pretty much locked into this very, very medial and stable origin. So really they grow outward and they get these big, huge, prominent pectoralis major muscles, stick out very, very obviously. And where does this muscle insert?
Intertubercular sulcus of humerus.
Yeah, that same intertubercular sulcus, right next to the latissimus dorsi. So while the latissimus dorsi is coming off of the back to the front here, coming off of the back and coming to the front, this one starts in the front and comes to that exact same insertion. And so notice it's going to move this arm in a very similar fashion. The pectoralis major is an anterior muscle. So being an anterior muscle, which way is it going to primarily move the arm?
Forward.
Yeah, and what do we call a forward motion of the arm?
Elevate?
No, it's decreasing the angle, so it is
Flexing?
flex. Absolutely. It is a medial muscle. So when it pulls the insertion towards the midline, what type of action is that?
Medially rotate.
Well, not yet. First, we're going to adduct, right? Being medial, it is going to adduct, so it's going to bring it in, right? But you guys are absolutely correct. Because it is an anterior insertion, it is when it's going to pull on the humerus, and it is going to medially rotate the humerus as well. So notice two of these actions, the adduction and the medial rotation, are exactly the same as the trapezius. Both adduct, both medially rotate, but the latissimus dorsi is on the back, so it extends, and the pectoralis major is on the front, so it flexes. So notice, because they have the same insertion, they are similar in their actions. Both adduct, both medially rotate, but the pectoralis major brings it forward, and the latissimus dorsi brings it back. So similar insertions, so similar actions, but one's on the front and one's on the back. All right. Questions on that? All righty. I think we just have one more muscle we need to do the origin on the insertion for, right?
Yes.
Yeah, the bad news is it's the tricep, which has 3 heads, the tricep brachia. So I think because of our time, I don't want to rush it. I think this is a good stopping point. We'll go ahead and stop here. We will pick up with that after the break, although I will strongly encourage you. So let's talk turkey here, right? You have 3 weeks till the exam, less than 3 weeks to the exam. This holiday break, while a chance to recharge, recuperate from all of this, is not time to take off. We come up and we're back and have a ton of material to cover, a ton of material to go over. Those are going to be 2 very, very long days, and then a weekend, and then the exam. So I want to strongly encourage you to be taking advantage of this holiday. Yes, take some time off, recuperate, recover, right? Say hello to your family that you haven't seen for weeks, but make sure you're using this time wisely as well. Use this time to help you to be successful. The advantage of this holiday is more time to synthesize this information. You have my lecture slides. You know exactly the things that I'm going to talk about for the rest of the exam. I mean, for the rest of the material for this exam. So there's no reason you can't start getting ahead and looking at the material. The advantage of looking ahead is getting some familiarity. In fact, one of the other students during my 5 for 5 told me that she's actually going back to to the fall's lectures to look at the lectures ahead of time. So if you want to look at the last 2 lectures ahead of time, see what I said last semester and look at that, absolutely do that. It can help you, even if you don't understand all of it. If you have some exposure to it before we talk about it in class, it can be something that can be incredibly helpful, right? The problem is you have to listen to the same jokes again, but because again, all my jokes are the same, but you got to remember, I tell the jokes for me. They're just to amuse me. If you get any joy out of it, then that's just bonus, but the jokes are from me. But things like that, looking at the lecture slides, looking at the previous lectures, those are things that definitely can help you to get ahead so that you're prepared, because the next test is going to come quick and we still have a ton of material to cover. All right, so again, take some downtime, relax, recuperate, recover, but also start preparing. Well, look, I'll be honest. I never had a YouTube channel until we went online. I encouraged students to audio record the lectures so that they had the things that I was talking about, but I never bothered putting together a YouTube channel. That seemed stupid. But it, because of the necessity of what we're doing here, I'm doing it. Honestly, when I get back on campus, after having doing this numerous times, I can't imagine. Well, the other challenge on campus too is, it's me and the whiteboard and the slides. So framing is probably an issue as well. So I don't think that it's something that I would continue once we're back on campus, but I don't think I'm gonna get rid of the lectures either. So having them to go to, if that helps you, then bonus. Again, so there are a few benefits to us being in this format online. So by all means, take advantage of all of them. All right. Any other questions? All right, you guys have a restful, a safe, but also a productive spring break. Don't go to Florida, and be safe, be healthy, be smart. And I will see you guys when we come back the following week.
I have a quick question.
No more questions. I'm done talking. Yes, what is your question?
You gave us some papers where we could draw muscle cells.
Yeah.
Is that just for us to kind of help us identify and learn? Is that something we turn in?
No, you're not turning. Great question. No, you're not turning them in. I've had, and again, this was even before we were online. I've had many students in the past who like, again, especially for very tactile learners, or visual learners, they like to take the pictures out of the textbook and actually label them themselves. I've had students who laminated them so they could write the answers and then erase them again, or whatever it is. So I think many students in the past have used it as a useful study tool, and so I've made that available for you guys. But no, there's no assignment. It's just another tool to help you to be successful.
Okay. Thank you.
You're welcome. All right, guys. Like I said, be safe, be happy, have fun, enjoy your week off, and I will see you April 6th. All right.
Thank you,
Bye.
Professor.
Thank you.
Thank you.
Welcome. Have a good break.